Good afternoon all, and welcome to the series of webinars from Sri Shankara Cancer Hospital, Bangalore. So today we have chosen the topic of HPV and immunology. So to start with, we will just go through a small prayer, which goes as Asato ma sadgamaya, tamaso ma jyotirgamaya, mrityorma amritam gamaya. So, as we all are aware, this is taken from Brihad Aranyaka Upanishads. It is a Shanti mantra, and it means that please, O Lord, take us from unreal to real or truth, from darkness to light or clarity or purity, and from death to immortality. So here we start our program today with a small introduction on HPV and immunology as to why did we take up this topic. I'll just screen share now. So as we are all aware, cervical cancer is the second most common cancer in women in our country. In the year 2020, it was estimated that around 96,000 of cases were recorded and around 60,000 of these women had died. It contributes to around 10% of all cancers that we see every day in our OPDs. But we also are aware on the other side of it. Despite knowing that this cancer is preventable and it has a vaccine and it can be screened and detected early, less than 3% of our women are coming forward to get it screened. We are in the process of educating them and vaccinating the children and screening our women. But there are some social and cultural factors that are hindering the growth of the decrease in these cancers. So today we have taken up this topic of HPV and immunology to discuss why and how only 1% of the HPV infected women develop cancer. What are the basis for various tests for HPV that we conduct on these women? And is there a role of hormones in carcinogenesis of cervical cancers? Can we use any anti-hormones as a treatment? And what is the basis of immunotherapy and its clinical problems? And what is exactly is this vaginal microbiome that we are reading of late in all the journals, and what is its role in carcinogenesis? So to start with, we have a panel of very eminent speakers, and I would request our director, Dr. Manjunath Shastri, to come forward and inaugurate this session.
Good morning, all of you. On behalf of the trustees and management of Sri Shankara Cancer Hospital and Research Center, I extend a warm welcome to you all. HPV infection, as was told by Annapurna, we all know that it plays an important role in the cervical cancer. And it's since recently, a lot of work has been done and the vaccination is on to prevent the cervical cancer. The topic of today, which was well thought and brought out by Annapurna and her team, appears very interesting. The microbiome of the vagina and the cervix, the tumor microenvironment, and the immunological response of the body to the infection, all these form a very interesting topics. With such eminent people on the panel who have enormous experience and expertise in their own field, I only look forward to a very interesting, thought-provoking discussions. I take this opportunity to welcome all the eminent speakers and Dr. Abraham, Dr. Maria, Dr. Smitha, and Dr. Jayashree, and Dr. Ravi Diwakar to have gracefully accepted the invitation to come forward and participate in this program and share their experience and expertise with all of us. Again, I take this opportunity to welcome you all and wish a successful program. Thank you. Namaskar.
Thank you so much, sir. And we would start our program and with Dr. Priya, none other than Dr. Priya. We all know Madam so well in this COVID times. And this Madam is going to address how our immune system reacts to this HPV infection and why Madam will throw light on the answer question that why only 1% of us get cancers, the ones who are infected with HPV. So as the Madam's CV goes this way, as after Madam had worked in Department of Clinical Virology, Christian Medical College. Madam was the chair of the department, and she had done various projects of DBT and ICMR and NIH and in her work on HPV. After and presently, Madam is the director and scientist cadre at ICMR and National Institute of Virology, Pune. Madam has had many firsts to her get credit. The first cases of SARS-CoV in India, first electron micrograph, first whole genome sequences, first virus isolation, first indigenous ELISA, first indigenous vaccine in partnership with BBIL, and the report of the first variant of concern in India were all done by Madam and her team. Madam also received Business Excellence and Innovative Best Practices Award, Academia Award 2020 from New Delhi Institute of Management, the Janani Award for Medical Research Leadership, and the Rotary Vocational Excellence Award, Harvard, and Sharada Shakti Award. So Madam had 176 publications to her credit. We are honored, Madam, that we to hear you today for this topic, interesting topic, which is very dear to your heart.
Thank you very much, Dr. Annapurna, for your kind introduction. And I would like to actually thank the director, Dr. Manjunath Shastri, Dr. Annapurna, other eminent faculty who have invited me to give this talk at the Sri Shankara Cancer Hospital and Research Center in Bengaluru. And I will attempt to talk about few facets of the immune response in HPV, which sets it apart from many of the other viral infections. I will now go to my full screen. And Madam had already spoken about the burden of cervical cancer. On a global scale, cervical cancer is the fourth most common cancer among women. And as per 2020, it has resulted in 604,000 new cases with 342,000 deaths on a global scale. And Madam had already alluded to it being the second most common cancer in our country, and also the amount of health burden it causes in our country. We do know that most of these cases and deaths come from the low and middle income countries in across the world. And we also know that 99% of anywhere between 95 to 99% of cervical cancer is attributable to HPV. And we also are aware that today HPV is rated as the most common sexually transmitted infection. Now, 50% of infections when women pick up infection are cleared by around 6 months. By 90 months, by 2 years, 90% of these infections are definitely cleared, and some are cleared over the next 1 year. When we say that there is virus clearance, it actually means that the virus is in a state of immune control, and maybe it may also mean that the presence of the virus is currently below the levels of detection. Cervical cancer, we all know, is the most frequent malignancy associated with HPV infection, where the precancerous lesions actually lead to cancer several years from the time of infection. So this image shows how the virus gains entry into the epithelium, the basal layers of the epithelium, and it is usually through breaks in the mucosa, minor injuries, that the virus gains entry. It gets into these basal epithelial cells and starts replicating at very low copy numbers. And these basal cells, as you know, slowly undergo differentiation. And by the time they reach the top, they are the typical flat squamous cell epithelial cells that are denuded from the epithelium. And as this virus migrates through these cells, which are migrating from the basement membrane over to the top layer of the epithelium, the virus initially expresses early proteins, later late proteins, and then the virus is assembled and then shed out from the top surfaces of the mucosa. When a virus infects an individual, most of the time, this happens 90 to 99% of the time. This happens almost unseen by the immune system, because the virus is mostly restricted to the epithelium. There is no viremia associated with this virus. And if at all the virus continues to persist, the normal epithelium goes through this switch over several years from a low grade to high grade epithelial changes, and finally to invasive cancer. This is a slow process, and therefore an infection such as this gives us an opportunity, wonderful opportunity, to intervene, one, in the form of vaccination, which is primary prevention, two, by meticulous screening. And that window of time is so long that we really in our country should harness that window of time wherein we can pick up the presence of dysplastic changes in the epithelium, as well as if we are applying a DNA detection assay, we could look for high-risk DNA and then follow up the individual and decide whether this needs further attention. Just to give you a little background, this is the genome, the circular double-stranded genome organization of this virus. This virus has an upstream regulatory region, which really has a transcriptional regulatory effect on these early promoters of these early genes. E denotes for early genes, and E1 is one particular gene that is involved with the viral replication. It's also described as a DNA helicase. E2 is another early gene that is involved with transcriptional control. It's tethering of the virus as an episome while the virus is replicating in the basal epithelium and migrating to the top layers. It also has a role in virus replication. E4 is involved with genome amplification and interaction with the cytoskeleton of the host cell. E5 also genome amplification and cellular proliferation. And E6 and E7 are well known to all of you. These are also known as oncogenes that express oncoproteins that are really the experts in viral evasion, immune evasion. These genes help in evading the immune response. And the last two gene segments or open reading frames are the L1 and L2, which code for the outer structural protein of this virus. So as I had described earlier, the virus gets in here, and you can see early in the course of replication, the early genes are expressed. And finally, while the virus is reaching the top layers of the epithelium, the late genes L1, L2 are expressed. Complete capsid assembly is over, and these are mature virus particles ready to be shed from the top surface of the epithelium. Now, I need to highlight that this virus has a unique life cycle. It infects the basal keratinocytes and epithelial cells via microabrasions. It leaves the basal lamina intact while it does all of that. Rest of the life cycle is mainly linked to the differentiation of the keratinocyte, as I told you. Virus assembly and mature virus particles are shed in the superficial layers of this epithelium. It is involved with minimal inflammation. This replication does not incite much inflammation in the site of replication. There's no lytic infection where the cells are lysed and viruses are released to within the epithelium. There is no viremia. It doesn't get into any of the capillary blood vessels in the mucosa. And the virus is shed from superficial layers quite distant from any of these vascular channels, thus not having access to any of the draining lymph nodes by virtue of entry into the microvasculature. Thus, the virus actually propagates with a very minimal, if any, inflammatory response. But nevertheless, the virus is very successful as an infectious agent, being capable of inducing chronic infection with no apparent systemic sequelae. Systemic sequelae will obviously draw attention to a lot more immune cells, and that doesn't happen normally. It rarely kills any of the host cells, and it doesn't kill the host itself, except when it goes on to cancer very many years down the road. It periodically sheds vast amounts of virus into the cervical canal, and can be transmitted to other individuals very effectively. And the virus cycle thus is a very effective immune evasion mechanism in itself. Now, one might ask, if this is such a clever virus, is the immune system at all involved? Does it play a central role at all in the control of this viral infection? And the answer lies in the evidences we see, particularly in the immunosuppressed population, where we see increased frequency of HPV-associated lesions in different types of immunosuppressed individuals. And I will detail that in just a bit, where you see increased detection of anogenital HPV, increased anogenital precancerous intraepithelial neoplasia, and also, if not detected in time, can go on to invasive anogenital cancer. Also, we see that when individuals are beneficiaries of primary prevention in the form of vaccination, a prophylactic vaccine, we find a very effective immune response, of course, prevents the chances of women picking up infection, and moreover, we do not see dysplastic changes in women who have been vaccinated early in their pre-adolescent years. And we all know the benefit that the anti-HPV vaccine has made in several countries, and hopefully will make bigger inroads in our own country with time. So with regard to what is the kind of immune response that is generated to this virus, there is something known as innate immunity, which is the first line of defense. There is, it is described as a non-specific response, but it's able to trigger the specific immune cells. This innate immunity essentially is doesn't have a memory, but it can trigger the main guys who can come to kill the virus-infected cells. This is done by the recognition of toll-like receptors, toll-like receptor 9. These are a family of receptors found in cells, and these toll-like receptors are able to activate signals to cellular to cells that of the adaptive immunity that I'll speak to you in a little while. And the main cells involved in innate immunity are macrophages, natural killer cells, natural killer T cells, and they're able to produce low levels of pro-inflammatory cytokines in the form of tumor necrosis factor. There is also TGF-beta, interferon type 1, alpha and beta interferons, etc. They are the trigger signals to the adaptive immunity, which may be in the form of cellular elements, that is the CD4, the helper T lymphocytes, the cytotoxic T lymphocytes, CD8 positive cytotoxic T lymphocytes, plasma cells, which are the factories of antibody and B cells. And these plasma cells are able to produce a good type-specific immunity, though in natural infection, this immunity, antibody-mediated immunity, kicks in a little late and may not be as strong as what we see following delivery of vaccine. But this adaptive immunity is, though the second line of defense, is exquisitely specific. It has an immune memory, particularly the T cells have a strong immune memory. Should a woman experience this, the virus again, these immune cells are able to come to the local site to contain infection if there is a second exposure. So this is the kind of immune response that HPV mounts. And if I were to show, in the epithelium are certain antigen-presenting cells. They are cells like big macrophages, which when there is any small amount of inflammation also, they get activated and they migrate to the localized lymph nodes, and they engulf some of the virus particles, present these particles to via major histocompatibility antigens type 1 and 2 to either the cytotoxic T cells, which are also known as the CD8 positive T lymphocytes, or to the CD4 positive T lymphocytes, which are known as the helper lymphocytes. And then what happens is these cytotoxic T lymphocytes migrate locally, they can infiltrate and clear the viral infection. Also, because these cells get activated, they can recruit natural killer cells and activate more antigen-presenting cells in locally, and also these are capable of sending signals for more of them to migrate to the lymph node and stimulate these cells of the adaptive immune response. Now, T helper cells are able to send cytokine signals to B cells, which will produce antibodies. Antibodies also locally can be used to resist further entry of virus particles. Remember, antibodies are not very useful if the virus has already gotten to the cell. But if there are free virus particles which have not yet infected the host cell, these antibodies, which are very type specific with some amount of cross-reactivity also, are able to resist further virus infection of cells at the basal epithelium. So the biggest evidence, as I had mentioned, that an immune response does exist in individuals against this HPV, despite its unique manner of replication, is evidenced by what HPV does in the immunocompromised host. In immunocompromised patients, HPV manifests a field effect. What I mean by that is HPV can be detectable in not only cervical mucosa, but vulval, vaginal, and can cause even in the anal epithelium to subsequently go on to progress to form dysplastic lesions that can progress to cancer. So because of the immunosuppression, HPV in fact has a more larger influence in the anogenital region, and many of these cancers are seen in those who are immunosuppressed. So this is a study, it's a very old study, but one of the seminal studies in India with smaller number of patients, but points to something very important. This even points to the importance of HPV-16 as one of the most, which is the most oncogenic of all the HPV genotypes. And this is a study that looked at the CIN and HPV infection in post-transplant patients in southern India. This was done at Christian Medical College, Vellore, where about 42 renal transplant patients and age and parity matched controls were recruited. This was the age group, and a prospective study was conducted over the next 2 years. The duration of immunosuppression varied anywhere between 6 months to individuals who were up to 8 years of immunosuppression, and individuals were followed up by taking a pap smear, doing colposcopy, and histological examination by a biopsy, and also doing an HPV-16 specific PCR. So in terms of the numbers who were HPV positive, these figures, these are the cases and these are the controls. They look somewhat comparable, but the odds ratio of people, those who had transplantation versus controls was about to be positive for HPV was to the tune of about 1.31. So there was definitely leaning towards those individuals who were immunosuppressed because of their post-transplant status to have HPV DNA in their lesions. Also, the duration of immunosuppression and percentage positivity for CIN lesions was seen. So those who were CIN positive were also studied for the duration of follow-up while being on immunosuppression. And you can see that at least, especially with at least 20% of individuals on immunosuppression, irrespective of duration of follow-up, at least 20%, which actually varied from 20 to 30%, had CIN positivity over time. We also looked at the histological level of CIN, and we could see that this is the distribution among those who were post-transplant and those who were controls. Obviously, in the post-transplant region, you can see clearly that immunosuppressed individuals who had had transplantation had much more CIN in them, and the odds ratio climbs up to about 6. Then I've put together some of the studies that are being published. There are not many actually, but and so these studies are not so recent, but nevertheless are seminal studies where they have looked at HPV and CIN in recipients of liver, pancreas, kidney, or kidney and pancreas. About 151 subjects were studied. The mean time between transplant and diagnosis of neoplasia was about 50 months, which is little over 2 years. All patients were found to be pap smear negative prior to transplant, and the incidence of HPV positivity after transplant was about 15.23%. And the incidence of the appearance of SIN23 or VIN2 or VAIN23 after transplant overall was about 7.28%. High-risk HPV was associated in most of these lesions, and lesions were multifocal and aggressive. And pap smear and HPV DNA testing is thus recommended before transplant and at least 6 months after transplant as part of regular follow-up in patients of these categories based on the findings we see here. I've also looked at bone marrow transplants. Again, there are not many recent studies, and the mean duration of follow-up in these post-BMT patients was about 31 to 42 months. Cytological abnormalities were significantly higher than an age-controlled population, odds ratio about 2.2, p-value 0.02. Abnormalities definitely were seen in post-BMT individuals with an odds ratio of 7. Allogenic recipients, those who had allogenic bone marrow transplant had a higher rate of abnormalities than those who had an autologous bone marrow transplant. And also in allogenic post-BMT patients, they had a significantly higher number of abnormalities as compared to the same group pre-BMT. So post-BMT was much more prevalent as compared to pre-BMT, odds ratio being about 6.8. Of course, we must remember that these individuals had other conditioning therapy that could have increased their immunosuppression using drugs like busulfan, etc. And we must also keep in mind that in post-BMT patients, sometimes the graft versus host disease may involve genital mucosal lesions as well, leading to dysplasia. I now come to an important group, not a negligible number in our country, where HIV-infected women have been studied for cervical HPV, and we have found that HPV infection was much more prevalent in women who are HIV positive. There are two major cohort studies in the US known as the HER study and the WIHS study, where it's two cohort studies that have looked at HIV positive women. And in these people, we could see that in HIV positive women, the prevalence of HPV infection was much higher, and there was a higher incidence of HPV in those that had lower CD4 counts, less than 200 per microliter, and those who had high virus loads of HIV RNA in them, typically meaning that with the higher levels of immunosuppression, these women were likely to be HPV positive. These women had multiple HPVs, high risk as well as intermediate and low risk types. A higher proportion of high risk types was seen in HIV positive women. There were also novel HPV types recognized, and very importantly, this has reference to my earlier comment in one of my earlier slides when we talk about viral clearance. Is it more about immune control? Because in immunocompromised individuals, there appears to be a reactivation of HPV infection and histological changes with advancing immunosuppression and with time. We also see that in the HER study, the predisposing factors for incident CIN was the presence of continued HIV positivity. HPV positivity was seen in those who had incident CIN, and like I had mentioned earlier, low CD4 counts, high HIV RNA levels, and this was the main risk factor for HPV to be persistent in women who acquired this infection in itself. Recurrent lesions were seen. They were more refractory to treatment, and low-grade lesions tended to progress to high-grade lesions more often. This is a study done in CMC Vellore about more than a decade ago. And this is, again, we were looking at HIV-infected women. There were three categories of women: those that are HIV positive themselves, those who were negative but had partners who were positive, those who were negative and who had negative partners as well. These were the two control groups versus women themselves who were HIV positive. And what this slide is trying to portray is that we looked at the single, dual, triple, or more than 3 types of different HPVs that were detectable in these women at a particular time of sampling. And we found that among HIV positive women, more of them tended to have not just mono-infection, single infection, but more of them tended to have dual, triple, and more than 3 different HPV viruses co-infecting these HIV positive women. In fact, a few of them even had 7 different HPV types detectable at one time of sampling. The other thing we found with the same group of women was that the risk of high-grade squamous intraepithelial lesions was again much higher in HIV positive women. You can see the odds ratio there, and the prevalence of high-risk HPV, again, even though they had a variety of HPV types in a single woman, the risk of high-risk HPV, odds ratio 3.2, was much more in HIV positive women. This is another study in HIV positive women coming out of Eastern India, where about 93 HIV positive women and a much larger number of HIV negative women were studied. They found that HPV 16 and 18 was very prevalent in HIV positive women as compared to the HIV negative women. And though these 2 types were very prominent, out of all HPVs detected, at least 53% were non-HPV 16, non-HPV 18. That means there were other types apart from 16 and 18. And also the occurrence of these oncogenic HPV types were not at all linked to the age of the patient or duration of exposure. So the fact that so many of these HPV infected women actually had so many non-HPV 16 and 18 is probably a flag for us, because both the current vaccines that we use in our country are directed against these 2 genotypes, not against the other genotypes, though there is some amount of cross protection within clades of these 2 high-risk genotypes. What is not shown in any of my slides here, I have also done HPV studies in men, HIV positive men who have sex with men. And you would be amazed that when we looked at anal HPV, these high-risk men who are HIV positive gay men, HIV positivity rate was to the tune of 94%. When we compared the penile and scrotal lesions, it was to the tune of 56%. And we also sampled the oral mucosa, because many of them are given to oral sex, and we could pick up HPV 26% in the oral mucosa of these high-risk HIV positive men. And again, many of these men their top 2 HPVs were not HPV 16 or 18. In fact, the more common were HPV 35, HPV 51, 52, and so on, then only followed by HPV 16. So then what triggers carcinogenesis? We spoke about the way the virus replicates mostly hidden from the immune system, unique manner of replication, but we have very strong evidences that when the immune system is down, we do see that the HPV is more persistent and can go on progress to neoplastic lesions. So the fact is that there exists a very delicate balance between virus clearance and virus persistence. And this is something very central, particularly to the carcinogenic potential of high-risk HPV genotypes, and may be attributable to the unpredictable cell division patterns in the stem cells found in the basal epithelium that HPV infects, may be attributable to the actual infecting high-risk genotypes, particularly if women have the high-risk types 16, 18, and the other well-known high-risk genotypes, may be linked to a weakened immunosystem, an altered hormonic milieu. Is it linked to the genetic makeup? We know certain genetic HLA proteins are associated with reduced risk for HPV. Some are associated with higher risk. And there may be other risk factors such as smoking and multiparity, which may all together contribute to why some women, thankfully a minority, a very small minority, progress to carcinogenesis. Now, how does the virus, if that is such a rare event, actually succeed in going on to cause neoplastic changes? And it lies in the fact that the two oncogenes, E6 and E7, code for two oncoproteins, as well as the E5 region, contribute to certain immune evasion mechanisms and pirating the whole functioning of the host cell in such a manner that it is skewed to virus persistence and reduces the cell senescence so that the cell almost becomes immortal. So if I were to start, the E6 through telomerase is able to cause almost replicative immortality. It resists the apoptotic, the normal apoptotic signals for cells, senile cells to actually die. E6 is able to subvert that. It deregulates the cellular energetics. E6 hijacks many of the important cellular genes. It can cause genomic instability and mutations. It can induce angiogenesis, which we know is associated with more risk for neoplastic changes. It activates invasion and metastasis. It also induces a tumor-promoting inflammation. It can avoid immune destruction by many of the cells by even reducing the number of major histocompatibility complex proteins to be expressed on the surface of dendritic cells and other important immune cells. And therefore, the cells of the adaptive immune response do not pick up the signals, and therefore we can actually, their immune response is curtailed. These oncogenes can even cause the higher circulation of T regulatory cells, which are well known to have an immunomodulatory, immunosuppressive effect locally in the epithelium, and therefore it can dampen the immune response locally. And these can also, as is well known to all of you, evade many of the growth suppressor genes such as the P53 and the RB genes via the oncogenes E6 and E7. So these are only some of the myriad ways by which these two oncogenes, if the virus is able to integrate and persist in the mucosa, subvert the immune responses which do exist in the epithelium, which can favor the dysplastic changes and movement towards cervical neoplasia. So what are some of the interactions with host genetics? We do know that some of the single nucleotide polymorphisms, they are known as SNPs for short, can lead to altered genome function and protein function. For instance, they're annotated like this. A SNP is typically annotated with some unique numbers that affect certain genes, so that when these SNPs occur, for instance, in the TGF-beta1 gene, this is associated with increased susceptibility to HPV-associated cancer. Why? Because TGF-beta1 actually is an immunosuppressive cytokine that impairs natural killer cell function. Remember, natural killer cell is one of the cells of the innate immunity, which is one of the early policing cells in the local epithelium. And this cytokine is able to impair, dampen NK cell function. Sometimes these SNPs can affect what is known as killer cell immunoglobulin genes, which are implicated in regulating the function of, again, NK cells and T cells. Sometimes SNPs in other genes. This is an important immunoreceptor, which again predisposes the individual to higher risk of induced cancer. So some of these interactions with host genetics can push a particular individual towards the risk of cancer. And as I had mentioned earlier, certain HLA or MHC genes, the human leukocyte antigen, or they're today most often referred to as major histocompatibility complex genes that code for certain proteins, are associated with a lower risk for cervical cancer, and a few have been attributed to a higher risk of cervical cancer. Of course, amongst us are one of the esteemed speakers, Dr. Kyrgiou from the Imperial College London, who would be talking of the vaginal microbiome and its association with cervical cancer. I've just touched on some of the other infections that could be associated with the persistence of HPV. And to summarize, I would like to say that HPV really does have a unique replicative cycle that normally would not provoke an intense inflammatory response. However, that immune response, particularly cell-mediated response, is critical to limit infection, as it most often happens, more often than not happens, as is evidenced by the impact of reduced immunity in individuals who are immunocompromised. This perhaps is the strongest evidence that immune response does play a role in limiting the progression of infection to become persistent and then go on to dysplastic changes. And high-risk HPV, we know, especially HPV 16 and 18 and 11 other high-risk HPV types, can successfully subvert the immune responses in several ways that could increase the expression of P6, P7 oncogenes and then evert the immune responses that would normally come into play to limit infection. And that is how lesions can progress. And with that, I would like to thank the organizers for inviting me to this important meeting, which has several important aspects of HPV infection. And I was also asked to touch upon a few possible research ideas that the Institute could take forward. So my ideas are very India-centric. So here are some of the possible research ideas that I would suggest: to look at the role of dry swabs in screening for HPV. This reduces the cost of getting the transport medium, and also it would be more rugged to environmental conditions and would not need storage in a refrigerator or later freezer. So this may be a good idea for a field-adapted study. You know, world over now, self-sampling at home has become the very popular way of enhancing cervical cancer screening. So I think this is something that the Institute should undertake in a particular community. We could also address in our own Indian situation, what are the barriers that come in the way of accepting HPV vaccination, cost apart. And then, of course, as I had mentioned earlier, HPV screening in high-risk women who have some form of immunosuppression. HIV positive women has been studied to quite a good deal in our country, both from Eastern India, Western India, Southern India, etc. But in unique populations such as those who have hematological malignancies and other malignancies who are on immunosuppression, it may be worthwhile looking at the presence of HPV, whether they are high-risk HPV types, do they persist, and following these women up to see whether that is associated with histopathological changes. And with that, I would like to thank the organizers of this today's program, particularly Dr. Annapurna, who has been tirelessly trying to reach out to me. And a big thank you to each one of you for your patient listening. Thank you.
Thank you so much, madam. It was a pleasure listening to you talk on HPV, and you have cleared so many of our doubts, madam. And I have a few doubts, ma'am. This HPV E6, E7 seems to be the main culprit. So we do have some diagnostics which will be coming up by Dr. Smitha will talk on it. Any antidotes or anything with herbal or HPV, any studies on that you had experience with, ma'am?
I'm so sorry. I have no experience with that, except that when you touched about E6, E7 base detection, actually some of the laboratories are, as you know, now we have a lot of platforms to detect HPV. Some people are going to detect the mRNA, not the HPV DNA, the mRNA of the virus, which denotes active replication of the virus. It's not merely there, but it is actually transcriptionally active. So that is known as RNA-based assays, which are associated with higher specificity and more likely to be associated with lesions that could progress. But unfortunately, I have no experience with any herbal remedies. Okay. I've done no such experiments.
There is a group from, if I may intervene, there's a group from Delhi. I think, if I'm not mistaken, it was Dr. B.C. Das's group, along with Dr. G.P. Talwar, who they published some work on neem paste being a very good antiviral agent.
Yes, neem has been explored. People have looked at turmeric, curcumin. Yes, there have been reports from other groups, but since the question was posed to me, I really I have no personal experience, and I'm sorry I didn't delve into literature and quote some of the studies. But I do believe that some of both neem and turmeric we know has got a lot of antimicrobial properties. They may be worth pursuing.
Thank you so much, madam. And you have very nicely elucidated to us as to how this virus, which is supposed to be very benign, like common cold, is causing so many numbers of deaths in our country. Very well explained, madam. Thank you so much, ma'am.
Thank you very much.
Now we go on to our next speaker, Dr. Smitha. Madam is the head of microbiology in our hospital, Sri Shankara Cancer Hospital, and she has vast experience working at St. John's and Fortis and Apollo hospitals, and she has attended and contributed to many papers and presented many papers in the conferences. Madam will talk today on what are these viruses, how do we detect them in labs, and what exactly is the mechanism behind these reports that we get in our clinics. Dr. Smitha.
Thank you, ma'am. Thank you for your kind words. Good afternoon, everybody. So at the outset, I would like to thank Annapurna ma'am and the whole Gynec Oncology Department of Sri Shankara Hospital for giving me this opportunity to become the part of the CME on a very important topic, that is HPV and immunology. So past 2 years, we have been listening to only one virus and its variants. And it's really nice to have, to speak about another important virus, that is human papillomavirus. So we know how important it is. And without much delay, I will go into my topic. So the topic which is given to me is HPV diagnostics. So when ma'am approached me for the CME, so she asked me, what do you want to talk about? So I said, being a diagnostic person, I would like to talk about HPV diagnostics. So she gave me a glimpse like, what exactly probably the audience want to hear from me on HPV diagnostics. So before that, let us just look into human papillomavirus. It's very important to know the structure of human papillomavirus for us to understand its diagnostics. So as you all know, human papillomavirus belongs to family Paproviridae and genus papillomavirus. So they're very, very small viruses, non-enveloped, has a single double-stranded DNA. So all viruses structures are very simple. They will have either DNA or RNA and a capsid. Some will have an envelope. So this virus is non-enveloped and it has a, see, this is the capsid. Okay, so it has a 72-sided icosahedral shaped protein capsid. This is very species specific, so that's why the name human papillomavirus. A lot of papillomaviruses, the one which is specific to human is human papillomavirus, and it infects the epidermis and mucous membranes of humans. So there are more than 150 types of HPV which are detected, and more than 40 types can infiltrate anogenital tract epithelium. And there are low risk and high risk based on the kind of diseases they produce. Low risk, benign lesions, high risk produces malignant lesions. Out of that, 16 and 18 are the most common one, which we are very much interested in. So coming to the genomic structure, I think Priya ma'am has already told in detail about the genomic structure and the pathogenesis, very comprehensive, very detailed explanation on that. But still, just to complete the topic, I would like to tell a little bit about the genomic structure. So it has a DNA which is circular, has 80,000 nucleotide base pairs, and genes coding for early proteins and late proteins. So we have 6 early proteins, 2 late proteins. So out of that, E6 and E7, as ma'am explained, are the viral oncogenes, which are very, very important for it to have a persistent infection in human epithelium. So again, I will not go in detail because ma'am has already told you about the pathogenesis. Just to give a glimpse of it, so there is persistent infection, which is very much required to cause a cervical cancer, or any cancer for that matter. Usually, HPV infections get cleared on itself in a matter of 1 or 2 years, but what will happen is they will evade the immune system, lead into persistent infection, which is a necessary cause of cervical cancer. So what happens when there is high risk HPV infection, which is persistent, there is high HPV viral load. So there is higher expression of oncogenes E6 and E7, which will act on P53 and rhabdoblastoma tumor suppressant gene, which is already explained. And this will cause deregulation of cellular proliferation. So the genomic instability which this causes, along with the environmental factors or other co-infections with other STIs, this will lead to HPV-associated squamous cell carcinoma. So this is the pathogenesis, what exactly happens in case of cervical cancer. So the studies which are done in India, they show that 98% HPV positivity in invasive cancer cases, whereas 20% in normal healthy controls. And we know that HPV 16 is the predominant type, while HPV 18 is very low, that is 13 to 15%. So these are the cancers which HPV 16 and 18 causes. So cervical cancer being the number one, almost 98% of cervical cancers are due to HPV infection. Very rarely, few people with no HPV infection can also get cervical cancer, but that is very, very rare. So coming to the WHO strategy, so it is based on this fact that 98% of these cancers are due to HPV infections. So it was endorsed by the World Health Assembly in 2020 that 70% of women globally have to be screened regularly for cervical disease with a high performance test, and for 90% of those needing it to receive appropriate treatment. So the summary of the recommendations, what they've done is for general population of women, screening has to be done with a HPV DNA. This should be the primary screening test, which should start at the age of 30, and every 5 to 10 is the interval they have mentioned for the general population. And for the women with HIV, the starting age of screening should be at 25, and the frequency of screening can be little lesser, that is every 3 to 5 years. So this tells us how important it is to have HPV DNA as the primary screening test. So my topic is on HPV diagnostics, which will tell you the different tests that can be done for screening purpose as well as for surveillance after treatment of CME lesions. So this is the clinical utility of HPV detection. First is screening of cervical cancer, and also in triaging women with atypical cytology, and after treatment of CME lesions, how it is progressing. So for these reasons, we do HPV DNA tests. So the finding carcinogenic HPV type does not provide a diagnosis of CM3 or cancer. We know that. It just identifies a group of women in whom CAN3 plus is more likely. That is the only thing. So it doesn't say that just because HPV is there, the patient is going to have cancer. So it is just, it will say whether the woman is going to get CN3 or cancer. The strength of HPV primary screening is its sensitivity. So when compared with cytology-based program, so which is less sensitive than HPV detection. So that is the strength. That is the reason why now everybody are recommending HPV DNA testing to be used as primary screening test. And another important factor is if the HPV test is negative, it is very useful. We can clearly say that the woman may not go in for CM3 lesions or cancers. So it has a very high negative predictive value. Coming to the laboratory diagnosis, so we know that traditional methods such as electron microscopy or cultures cannot be done for HPV. Even the immunological methods, serological methods, basically because it is localized infections to the epithelium, right? So it doesn't come into blood. So the serology detecting antibodies is not very helpful for us here, which is done for other viruses, right? So the molecular detection is the only answer to identify HPV. The clinical specimens which are used are endocervical cells, right? So this is obtained by cervical brush. Also the cells in liquid-based cytology sampling can be used with thin prep, okay, which are collected with a broom type or cervical brush. And cervical biopsy specimens can be used for in situ tests. As ma'am told about self-collected vaginal samples, so it has become increasingly popular now. Okay, women prefer taking their own samples than going to a clinic and taken by a practitioner. So, but what we have seen that there are different tests available. So the ones like which are based on signal amplification, like our hybrid capture or CARE HPV, are found to be a little less sensitive and specific on these samples than PCR-based assays. So PCR-based assays have very good sensitivity and specificity, both for cervical as well as vaginal swabs. Urine is another sample which has been thought about, but the sensitivity is very, very, very low. So right now we cannot think of urine as a sample for HPV screening. Coming to the molecular methods, so we have non-amplification hybridization techniques, and then we have amplification techniques, which are either signal or target-based amplification. So the non-amplification nucleic acid hybridization assays are southern blotting, in situ hybridization, and dot plot hybridization. So out of these, in situ hybridization still have some clinical value, but other two have not been pursued much. Basically because of the low sensitivity. It is the sensitivity is less than 89%. And the need for relatively large amount of purified DNA. Whereas in PCR, where we are amplifying the DNA, so we get sufficient amount of DNA, even 1 or 2 is sufficient for us. But here, a relatively large amount of DNA is required to identify. And it is time consuming and requires skilled personnel. So in situ hybridization probably still has clinical value, basically because it allows simultaneous identification and localization of HPV infected cells. Okay, so this is the advantage, and also the signal pattern which it gives. Okay, so it is more relevant for detecting CAN3 lesions. So that's why this has little more advantage over other hybridization assays, and the sensitivity is around 89%. So this is how it looks. So you can see the punctate form. This is the signal patterns I was telling. Either you can have a diffuse form or you can have a punctate pattern. So the punctate patterns give more this thing towards CAN3 lesions. So if we have punctate, that means the lesions are CAN3 plus. So that is the advantage of having FISH assays. So as I told you, with the exception of FISH, which continues to have many uses, non-amplification methods have been currently replaced by amplification methods. So amplification techniques are the ones which are favored against these non-amplification methods. So there are signal amplification methods, there are target amplification methods. I will go in detail about these tests so that you will have an idea on the sensitivity and the predictive value of each of these tests. And depending on the target, as ma'am told, either we can have DNA detection, which is very, very common. Most of the assays do detect DNA based on L1 region or E6, E7. And there are some assays on mRNA detection, which will tell about the E6, E7 oncoproteins. So these are the different assays which are available. So coming to the hybrid capture assay, which is a gold standard in HPV diagnostics, and it is used as a comparator assay. Means any test which have been which requires new validations have to be compared with a comparator assay. So hybrid capture is one of that assay. There are other two assays which are comparator assays, but this is the gold standard, and most of the people use this as a validation technique. Okay, so it's a signal amplification technique just for the academic purpose for the students here. So I'm just going to explain how exactly it happens. So HCV has double stranded DNA. So you can see this is the double stranded DNA. So first we need to denature, make it single stranded. Then we have HPV specific RNA probe, which gets hybridized with DNAs. Okay, so this is the RNA DNA hybrid which is formed, which will be captured onto a microplate well, which is coated with antibodies against these hybrids. Okay, so they get captured here. So that's why it is hybrid capture. Okay, so once it gets captured, so we add these antibodies with alkaline phosphatase enzyme. Okay, so they come and get attached to these hybrids. Okay, so when we add a chemiluminescent substrate, the substrate, this alkaline phosphatase enzyme, will act on that and it will give a light. So that is how we detect. So the signal amplification is happening here because the number of antibodies which get attached to the hybrid. So there is no target amplification. So it's basically the signals which are amplified here. So this is how we detect in hybrid capture assay. So there are two assays. One that detects 5 low and 13 high risk types, and another one detects only 13 high risk types, which is the most commonly used. But this is only for detection of HPV. We cannot specify the genotypes here, right? So that is the thing with hybrid capture 2 assay. There's no internal control which will tell the sample integrity. So there's no internal control which is used here in this test. The limitations are it is qualitative or it can be used as a semi-quantitative assay based on the RLU, that is relative light units per cutoff. Okay, so based on that we can say that if it is more than 2, it favors CAN2 plus. So as per the kit inserts, more than 1 RLU is positive for HPV, but if it is more than 2, we can think that it is probably into CAN2 plus lesion. The disadvantages, yes, as I told you, it doesn't specify the genotype. And there are some cross reactions which are seen between high risk and low risk types in hybrid capture, one of the assay, and it has to be run in batches of 98. and it takes around 5 to 6 hours for each run. So we have to wait for the batch to fill in, and then only we can run the test. So that is the disadvantage of having a hybrid capture to assay. The sensitivity is around 85.7%. This is all for predicting high-grade squamous intraepithelial lesion. Okay, the sensitivity is around 85%. Specificity is low; it's around 67%. The negative predictive value is really good, 99.6%. Positive predictive value is 4.5%. So we cannot say that all the lesions are going to go into CM3+ or cervical cancers. But this is a screening test, and we know that screening test has to be more sensitive than specific, right? And the detectable limits are 0.62 picogram ml, so around 1,000 to 5,000 copies per reaction. So that much should be there for it to detect. Another one important test is CARE-HPV. So for developing countries, QIAGEN has come up with this CARE-HPV, wherein it's difficult to use hybrid capture 2 technology. So this CARE-HPV has the same principle as hybrid capture, okay, but it takes around 2 hours 30 minutes, and it is designed to be run in developing less developed countries with minimally trained individuals. So that is the advantage of having CARE-HPV. Other than that, sensitivity is little low, 87%, and negative predictive value also little lower than hybrid capture. It's around 91.11%. Other signal amplification method is Cervista, which also detects the genotype 16 and 18. Okay, so this has been approved for screening with women with ASCAS and in conjunction with cytology for patients above 30 years to determine high-risk types of HPV. It is based on an invader technology and has 2 isothermal reactions. It can detect up to 1,000 copies, okay, and takes around 5 to 6 hours for each test to be done. So just for an academic interest, so this is how the test is done, hope you can make out. So this is the target DNA which we are interested in identifying. So what happens here is we add a probe, okay, which is specific to this, the sequence is specific to this target DNA, and also we add an invader. So this is an invader oligo. So what happens here is there is one base. Okay, so here you can see one base pair overlapping, which is happening here. Because of this, it gets cleaved. Okay, so the probe which we have added gets cleaved and it comes out as a separate flap. So this is one reaction. In the next reaction, this flap will act as an invader. So there is this fret probe. Okay, there is this fret probe which has a fluorophore and a quencher. So when this fluorophore is with the quencher, there is no fluorescent signal which is seen. But when this flap gets attached to this fret probe, okay, again one base overlapping happen, because of which another cleavage happens here. The fluorophore comes out of this quencher and it gives a fluorescent signal. So this is the principle of this Cervista test. So this is how we detect the signal. So again here the target is not amplified. It is the number of signals which we are looking into. The sensitivity is around 92%. Negative predictive value is 99%. This is for predicting HSLs. The advantage is it includes low or no cross-reactivity with low risk HPV genotypes. So hybrid capture had some cross-reactivity with low risk HPV tags, but here there is no cross-reactivity, and there is an internal control which is added. So it will tell us the integrity of the sample, that we have taken good amount of or good quality sample. So that identification we can do with this test, and it requires the sample volume required is low. In comparison with hybrid capture 2, it has got 100% sensitivity in the detection of CAN3 and 98% in the detection of CAN2. Also, it has lower false rate and high sensitivity. PCR. So PCR is the method which we say, right, about amplification. So everybody knows about PCR. thanks to COVID, I think even common man knows about RT-PCRs nowadays. So we can make a number of copies of a single DNA segment. So that is the advantage of having a PCR. So just to give, everybody probably knows, but just for an academic purpose for the students, I'm going to tell this. So what we do in PCR is we take the double-stranded DNA, we denature it, make it single-stranded DNA at the temperature of 94-95 degrees centigrade. Then we add the primers, which are specific to the targets which we require, that is annealing stage at 50 to 56 degrees centigrade. Then we extend it at 72 degrees centigrade, and that is how we amplify. So this go on amplifying. So a single copy can be amplified to 1 billion copies by the end of 30 cycles. So that is the advantage of having a PCR technique. So the primers targeted in HPV are L1 viral capsid. Okay, so that is the major target we use for PCR. And also some have type-specific PCRs, which are where we use primers against L1 and E6, E7. There are some tests available with L1 and E6, E7 targets as well. And the advantage is HPV genotypes can be determined separately. But for PCR, we need other techniques to determine the genotypes, that are restriction fragment length polymorphism, linear probe assays, direct sequencing, or genotype specific primers. So any of these methods are used to identify the amplicon from PCR. The sensitivity is markedly increased here. Okay, it can identify as few as 10 to 100 HPV DNA molecules. So that is the major advantage of PCR. Sensitivity, you can see it's very good, 95%. And negative predictive value also really good, around 99.8%. This is for predicting HSL. Real-time PCR, the methodology is same as PCR. The only thing is we are adding fluorescent probes with PCR primers so that we can identify the genotype and the HPV in the same reactions. In PCR, what we are doing is we were amplifying, and in another technique, we were trying to genotype, but here it happens in the same reaction. So that is the advantage of real-time PCR. The sensitivity remains same as PCR. The advantage is we can estimate the viral load. So how much virus, the quantification can be done here, which is helpful in identifying the persistent HPV infection. So that is the advantage of real-time PCR. And a little bit about mRNA detection. So ma'am was asking about E6, E7 specific tests. So there are some tests which will identify or which will help in identification of transcripts of E6, E7. So basically they detect the mRNA for E6, E7. Okay, so it will increase the specificity. As ma'am was telling, mRNA implies that there is active viral transcription. So viral DNA means the virus is present, but it may not be actively transcribing, right? But here we can know that active viral transcription is happening because we are detecting mRNA, which is a factor required for viral transcription. So that is the major advantage of having mRNA detection tests. So the specificity really increases. So there are 3 different assays as of now, which are developed for mRNA detection. PreTect, which is on NASPA, based on nucleic acid sequence-based amplification. Here we can detect up to 5 high-risk types. All these will include 16 and 18, along with the 2 or 3 more other high-risk genotypes. Then there is Aptima, which will detect 14 high-risk types. But here also, we cannot specify which genotype. It will just say whether these genotypes are present or not. This is based on PCR method. And then we have RNA flow cytometry technique with fluorescent probes, okay, Oncotech, which will detect again E6, E7. And this is in a research process. So still they are trying to find out how good it is in the clinical studies. So there are a number of articles on this comparison between DNA and mRNA testing. Okay, so one of the article I saw, and it was very interesting to know that the sensitivity of mRNA and DNA for CN2+ and 3+ It's almost same. Sensitivity is not much different, but when it comes to the specificity, you can see mRNA is around 79 and DNA around 71 for CN2+, whereas here also you can see for CN3+, the specificity is more with mRNA than DNA. Okay, and the positive predictive value also is little higher with mRNA for both CN2+ and 3+. Whereas negative predictive value is quite similar, around 95, 96 for both mRNA and DNA. So we can see that by this study that, mRNA has more specificity and more positive predictive value than DNA test. But there are not many kits available for this, and studies, multicentric studies have to be done to understand it better. So coming to the benefits and weaknesses of these methods. So we have nucleic acid hybridization says because of low sensitivity and the time it takes, it is not used nowadays, whereas signal amplification and nucleic acid amplification assays are used. So signal amplification is a semi-quantitative method, right? So it will tell us whether the patient is in CN2+ or 3+ and has lower false rate, but individual genotyping is not done here. So we will not know which genotype it is. Whereas with nucleic acid amplification assays, they are very, very sensitive, and you can give quantitative, you can exactly tell how much viral load is present. Multiplex means it can detect n number of genotypes. The weaknesses are some genotypes will have lower amplification signal and false positivities are high with this PCR method. So these are the weaknesses of nucleic acid amplification assays, but still they are considered better for screening. So I found an article which has listed down all the tests which are available, right? So many tests are available for HPV DNA and mRNA detection. Okay, and they have done a validation study in the year 2020 to find out which are good in terms of sensitivity, in terms of reproducibility between inter and intra-laboratory comparison. So this hybrid capture and GP5+, 6+ based PCR-AA, they are the comparator assays. So they have done the comparison of these other tests with these two, any of these two, okay? So they are the comparator assays, and we have about COBA. So like so many are there. So the ones which have colored in green have shown good validation result, means they had very good reproducibility with inter and intra-laboratory comparison. The sensitivity and specificity were really good with these tests. About COBAS, Alinity, expert HPVs, all these are really good. So these are targeting E6, E7, whereas others were targeting L1. So these are the targets genes which are used. Okay, so you can see the amplification types. So most of them are target amplification tests. And here you can see the sensitivity, almost all are above 95%. So these, the ones which are in this purple color, they are there in the market, have shown good sensitivity and specificity. But when it came to reproducibility between interlab and intra-laboratory comparison, they did not show the same reproducibility. So that's why these are not, probably we need further studies to actually validate them and see how we can use them. So, but based on these multicentric studies, they have taken a lot of multicentric studies. Based on this, they have chosen these 7 tests as more validated tests which can be used for HPV detection. Okay, and these are the mRNA kits, okay, which are available based on flow cytometry, target, NASPA. But we require lot many. Aptima is still more commonly used, which has been done, which a lot of studies have been done on Aptima, but we need to have a lot of studies on Oncotect and Pretect to know whether we can actually use them as a screening method. Coming to the viral load. Okay, so this is very important and it can be determined by real-time PCR techniques. studies by Lowe et al. He has demonstrated that there is significant difference in viral load between CAN1 and 2 and between normal and all CAN, but not between 2 and 3. Okay, so this is very, very helpful for us, because if there is a significant viral load, it means that the patients are going for CAN lesions. Okay, so this is the utility of having a viral load testing done. So it basically predicts the progression or severity of the disease. And also in response to therapy, the viral load decreases. So these are the advantages of having HPV DNA viral load testing done. And the utility of testing HPV DNA integration. So we know that the HPV DNA has to be integrated into the host cell so that it can have persistent infection, which will lead to cancer. So the viral integration is a very important step, and an early event occurs earlier than the morphological changes. Before any cytological changes happen, the viral integration is happening. So if we can identify it at that time, so it actually helps us to screen them better. Okay, so the main methods used for integration are PCR, FISH method, and real-time PCR, which actually gives the quantification. PCR uses E2 type specific, which is for replication of the virus, okay, and E1 type specific primers. So these are used, but studies have to be done on the techniques to prove how effective it is going to be. But theoretically, it looks really good that, if you can find out if there are tests to identify the HPV DNA integration, which is required for the development of cancer, it actually helps the treating doctor, the clinician, to screen the woman better, right? So HPV DNA viral load quantification, integration, E6, E7 expression are promising biomarkers that can predict the progression of lesions to cervical cancer. So this is the glimpse, this is the summary of the test which I have told. There are some newer isothermal amplification techniques which are coming up, which are simpler, which are faster, and does not require high-end reaction because they act in one constant temperature. As I told, PCR is on thermal cycles, so you need to have different temperatures. So we need to have high-end instruments, which are a little costlier. But here in these cases, we can have a simple machine, simple instrument, because they are isothermal amplification. So here we will be using enzymes to amplify the DNA, target DNA, right? Here we are not using temperature as the factor for denaturing the DNA. We are using enzymes. So there are loop-mediated amplification method called LAMP. Okay, it detects up to 7 high-risk types in a matter of 1 hour. So this is very, very, attractive. The time taken is faster, and it can detect as low as 10 copies per reaction. So basically an amplification method, but in an isothermal way. Okay, and there is CRISPR-Cas12a technology. So this method also is used. So there are some tests available which are in the research areas. They're doing the studies, clinical trials are being done. It takes around 35 minutes. Okay, it's very, very attractive, and it can detect up to 100 copies per reaction. So all these are newer methodologies which we are waiting, okay, so that it can come into the market, and it will be helpful for screening mass, in mass cases. So very simple and faster reactions. About anti-HPV antibodies. So antibodies are not useful much in detection of HPV because the sensitivity, you can see, is very, very low. 53%, 67%, because it's more of a localized infection, right? It happens in the epithelial cells. It is not coming into blood for us to detect the neutralizing antibodies. But there are some assays available. There are some studies which are done on a virus-like particle 16 and E4/E7 antibodies with very less sensitivity. Specificity we can say is 97%, basically because that has to be there for the antibodies to be produced. The specificity is really good. And there are some new point of care biomarker tests based on antibodies to HPV16 DRH1. So this new test also will detect an antibody which is produced only when an infection has led to increased cell growth. So we have to see how these studies on these new biomarkers are going to go and whether we can use it for our HPV detection or as a screening method. So here ends my topic. So these were the references. And I would like to thank Annapurna ma'am and all the organizing people here who have given me this opportunity to talk on this HPV diagnostics. As I told, it was really pleasure to talk about something other than SARS-CoV-2 in the past 2 years. So we have been talking only about that. So thank you, ma'am. So if any questions are there, I would like to take it now.
Thank you so much, Smitha. It was wonderful listening to the basics of the test that we ask for. There's one question by one Dr. Shiv Kumar. He's asking, HPV mRNA detection tests, do they take place in Bangalore, and what is its cost? Any idea about that?
I need to look in, ma'am. Basically, in Bangalore, what we do is DNA testing. So we have not come up with mRNA testing. There are a couple of centers which we do. I mean, probably we can send out the samples there and find out. Or actually, we can actually, as a research module, we can start that mRNA detection and compare with the DNA detection. But as of now, in Bangalore, we are not doing this.
None of the labs in Bangalore are doing mRNA testing.
Yeah, most of them are doing DNA detection.
Now, further questions I can see on the chat box. So can we move on to the, thank you so much, Smitha.
Thank you, ma'am. Thank you, ma'am.
Now we move on to the third topic of the day, and that is by Dr. Jayashree madam. She is going to talk on the role of hormones in cervical carcinogenesis. This is a new topic that has come up, cancer and cervical cancer and hormones. We have always been listening to cervix, in hormones and endometrium or breast, but cervix, though it is very close to endometrium, still what is its present role? We need to get information from madam. Madam is PhD from PGI and IUCC fellow, and madam had served in Kidwai, Bangalore for 27 years at various positions, and she retired as HOD professor and HOD of Department of Microbiology in Kidwai. Madam is a recipient of national and international awards like ICRET Fellowship, IUCC Fellowship, IR, and Travel Awards from DOT Keystone Global Health Symposia. Madam had done as a principal investigator or a co-investigator on 15 projects. And whatever I have listed here are basically, I have taken for the ones that she had worked on cervix. This will give an information as to her capability of as a researcher, and that madam has contributed to literature so much. She has done studies on feasibility of multifunctional nanoprobes for cervical cancer therapeutics, HPV and other etiological factors in head and neck cancers, genomics of cervical CAFs and immune tolerance, and regulatory T cells as marker of progression for head cells, and TLR expression profiles in cervical dysplasia patients, and development of HPV vaccine prototype and HPV types that are present in India, and its identification of 16L1 and E6 variants, and molecular diagnosis of common opportunistic infections in these women, cervical cancer women. Apart from this, she has also had past experience of work on Epstein-Barr virus, toxoplasma, and HPV. Madam has 52 original articles to her credit. So here we are pleased to invite Dr. Jayashree madam to talk on hormones and the role of hormones in cervical carcinogenesis.
Good afternoon, everybody. Am I audible?
Yes, ma'am.
Good afternoon, everybody. At the outset, I wish to thank the organizers, Dr. Annapurna and Dr. Srinath, for having invited me to be part of the CME. Now, can I have the first slide, please? This is not the first slide. I want the first slide. Next. Yeah. So as a prelude to my talk, I wish to first introduce some aspects of the tumor microenvironment, because I think some of these aspects may be new to at least some people in the audience. So any solid tumor is basically, it comprises of two distinct, poorly demarcated areas. One is the parenchyma or the tumor bed, which most of you are familiar with. And the second thing is the tumor stroma or the tumor microenvironment. So basically the tumor microenvironment, it comprises of non-malignant cells, basic blood and lymphatic vessels, mesenchymal stem cells, cancer-associated fibroblasts, pericytes, infiltrating immune cells belonging to both the innate and adaptive immune systems, the extracellular matrix, and secreted soluble products. Next, please. Next slide, please.
Yes, ma'am.
Yeah. So the TME is basically the environment in which the tumor cells are surrounded. So this TME is basic. You can compare it to the roots of a tree. It plays a crucial role in the development, growth, and evolution of the tumor. And there is a constant, dynamic, bidirectional crosstalk between various components. And in the case of cervical cancer, you have three components: HPV infected epithelium, the stroma, and infiltrating immune cells. With all these three together, they play the interaction between these three components that plays a major role during carcinogenesis. And so since there is a constant dialogue occurring between these components, so the final net result of tumor elimination or survival will actually depend on which side the scale tilts, whether it tilts towards pro-oncogenic, in which case the tumor will survive, or whether it goes towards anti-tumorogenic activity, which factors will gain an upper hand. That will depend on whether the tumor will survive or get eliminated. Now, to elaborate a little bit about the various components of the tumor microenvironment, you have pro-tumorogenic cells, that is basically cells which will suppress the immune response locally. And that is like, so there are various cells, and I would like to define some of these components a little more in detail, because I'm going to highlight or going to talk about the work that we have done on these cells. So Tregs or regulatory T cells, what are these? So when I was a student in the 1980s, we were taught about suppressor T cells, which were CD8 positive. Later, the concept died down because nobody could show the presence of these cells. Definitely, they couldn't demonstrate these CD8 positive suppressor T cells properly. And then, so the labs which were working on them, they closed shop. Some people committed suicide, and then the whole concept died down. Later in the 1990s, Dr. Shimon Sakaguchi actually demonstrated, or rather resurrected, this population, but called them regulatory T cells, with the difference that these were CD4 positive, CD25 high, that is They expressed IL-2 receptor high, and they were FOXP3 positive, which was the marker and a master regulator of regulatory T cells. So these regulatory T cells, which are CD4 positive, CD25 high, and FOXP3 positive, they are one such dominant population within the tumors which suppress the immune response. Another population is myeloid-derived suppressor cells, M2 subtype of tumor-associated macrophages, and carcinoma-associated fibroblasts. The anti-tumorigenic armamentarium comprises of CD8 positive cytotoxic T cells, dendritic cells, Th1, Th2 subtype of CD4 T cells, natural killer cells, M1 type of tumor-associated macrophages, and so on. So these are basically effector populations which will actually perform the job of killing the tumor cells. Now, in addition, you have pro-tumorigenic molecules, which are immunosuppressive cytokines like IL-10 and TGF-beta. And you have some immunosuppressive molecules also, which you must be very familiar with, at least after the advent of checkpoint inhibitors. CTLA4 and PDL1 has been introduced, and people are aware of these. You also have anti-tumorigenic molecules, that is cytokines of like gamma interferon and TNF alpha, and molecules like perforin and granzyme, which are secreted by cytotoxic T cells and natural killer cells and perform the job of lysis of the tumor cells. Next, please. Can I have the next, please? Yeah. So we and the others have been working on the intratumoral immune response in cervical cancer. And we see, so what we have learned over the years, that the infiltrate in cervical cancers, it doesn't lack effectors. So it's not that there are no effector populations of cells within the tumor. You have CD4 positive effector T cells, which are both of Th1 and Th2 subtypes. You also have HPV specific cytotoxic T cells, NK cells, Langerhans cells, dendritic cells, M1 type subtype of tumor associated macrophages. Some of these, like Langerhans cells, have been shown to be anergic. However, the others, CD4, Th1 and Th2 subtypes, they are not anergic, but however, there are these, because of the suppressor populations of T cells and other suppressor cells, these suppressor cells, they somehow dominate the show, and they do not allow the effector populations to perform their action. Next slide, please. No, the previous one. So a little bit about, Dr. Priya has already spoken in depth about cervical carcinogenesis initiation, at least. So we all know that high-risk HPV infection is a well-established cause for cervical cancer. But for a full-blown invasive disease to happen, HPV needs to be assisted by various cofactors, some of which are the viral load of HPV, genotype of HPV. We all know that 16 and 18 have a more oncogenic potential, and that is why they are known as high-risk HPV types. Smoking, so exposure to tobacco in the form of smoking. Other sexually transmitted infections, Dr. Maria will be speaking later in the day, and she will tell you more about elaborate more on how they do it. But anyway, I suffice here to say that sexually transmitted infections, one thing that they do is they make it possible for HPV to access the basement membrane, where you have these stem cells where the virus can enter. And exposure to estrogen, which has been considered a co-carcinogen in HPV-associated cervical carcinogenesis. Now, to highlight the fact of estrogen being an important cofactor, there have been, multicentric large epidemiological studies which have been carried out across continents, which has shown that multiparity with more than 6 full-term births increases the odds ratio of a woman, HPV infected women, to develop cervical cancer 5-fold. Similarly, oral contraceptive use for more than 5 years increases the chances of a HPV infected woman to develop cervical cancer 3 to 5-fold. In addition, a large prospective Danish study recently which has been published, showed that women diagnosed with cervical cancer during pregnancy had a higher risk of dying from the disease due to elevated plasma estrogen levels attained during pregnancy. Next, please. So, there were two important points which actually led us to get interested in this topic. So one is that there are no animal models for HPV, human papillomavirus mediated cervical carcinogenesis. However, there are some animal papillomaviral mediated models wherein people have used these models to elaborate various aspects of host virus relationship and so on. And one of which is that the K14 HPV 16 E6 E7 transgenic mouse model, where the HPV 16 E6 E7 has been, by recombinant DNA technology, K14 promoter has been used to insert the HPV E6 E7 component of the viral genome. And in this animal model, they have shown that this animal model is based on the requirement of both estrogen and estrogen receptor alpha signaling for the disease to evolve from simple infection to full-blown cancer. So this animal model and the epidemiological studies which I've just indicated in my previous slide, which showed linkage between estrogen exposure and cervical carcinogenesis, prompted us to investigate the role of estrogen as a cofactor in HPV-mediated cervical cancer. Next slide, please. So I just want to highlight what estrogen does in cervical cancer. Basically, before we come to the point of cancer, what estrogen does in the cervix. So uterine cervix is an estrogen responsive epithelium which proliferates and differentiates physiologically during the menstrual cycle. And so estrogen has got the capacity to act on a cell using two mechanisms. One is the genomic pathway and the second is the non-genomic pathway to signal the epithelial cell. So genomic pathway utilizes the estrogen receptors, various estrogen receptors, estrogen receptor alpha, beta, G protein coupled estrogen receptor, etc. So in the canonical or genomic pathway, what happens? Estrogen first combines with estrogen receptor alpha, then the complex of estrogen receptor complex, gets translocated to the nucleus, and in the nucleus it goes and binds to estrogen responsive elements on the DNA. Here epigenetic modifications take place and the gene finally gets activated. Now a lot of this work, a lot of the knowledge, almost the entire knowledge about estrogen pathway has come from our understanding of estrogen as a carcinogen in breast cancer. So, there are various estrogen receptor antagonists, selective estrogen receptor modulators like tamoxifen, and selective estrogen receptor dysregulator or disruptor like ICI 182780, which is nothing but fulvestrant. Both these have been used in the past as treatment for breast cancer. So I just would like to mention here that please bear these in mind because I'm going to talk about it a little later. Now, in addition to the genomic pathway, estrogen can also signal a cell through the non-genomic pathway or the non-canonical pathway. And this is because of its ability to do a molecular crosstalk with various other growth factors like insulin like growth factor, epidermal growth factor, and fibroblast growth factor. So in a way, irrespective of whether a cell once again, I want to stress here. So irrespective of whether an epithelial cell has an estrogen receptor or no, the cell is going to get affected by the presence of estrogen in the environment. So this is one thing which estrogen does. It signals an epithelial cell towards proliferation, differentiation, etc. And in addition, if there is estrogen in the microenvironment of a tumor, it can also act both by the genomic and the non-genomic pathway to modulate various cells in the tumor microenvironment, and hence in this way also promote tumor growth. Next slide, please. So this was the basis which led us to get interested into the topic of estrogen and cervical cancer. So we, as a first step, we wanted to see whether, it's okay when we are talking about a mouse model of cervical carcinogenesis, but in actual principle, do we really see heightened levels of the hormone within the tumors? So what we did was, we decided that we would estimate 17 beta estradiol in the plasma and cervical tumor tissue specimens. So we collected blood and about 100 milligrams of tumor tissue, the tissue adjacent to the tumor, from cases of squamous cell carcinoma FIGO stage 1B2. And I'm thankful, very thankful to the resident pathologists of the pathology department of Kidwai under the guidance of Dr. Jayashree Mukherjee for having made this possible for their dedicated sampling. Now, blood and cervices from age-matched women undergoing hysterectomy for benign diseases served as controls. Estradiol was extracted and assayed biochemically. Here, I wish to thank Professor A.J. Rao, Emeritus Professor of Biochemistry in IISc, who trained my graduate student, late Dr. Srinivas Adurthi, in this process. So what we found was that the estrogen levels in squamous cell carcinoma was 3 to 4-fold higher than benign cervix, meaning normal cervix from benign disease. And even the adjacent tissue had levels higher than the benign cervices. So this indicated that, field cancerization was happening, is one additional point which I would like to mention here. And the surprising part was that the plasma estrogen levels in the patients with cervical cancer was in fact much lower than the plasma levels in the patients with benign disease. Next, please. So this actually meant high estradiol in the tumors either meant that there was augmented absorption and retention of circulating estrone or estradiol. However, from our study, since we found that plasma levels of patients was much lower than even the controls, it obviously meant that plasma levels of estradiol was not heightened. That means that there was more likelihood that local production of estradiol was happening. So estradiol is produced by aromatization of testosterone. Androstenedione is a precursor of estrone. Aromatization of androstenedione gives rise to estrone. And estrone is actually normally it is found as a latent form, estrone sulfate, in the tissues. So estrone sulfate can get converted into estrone by steroid sulfatase, and this estrone can actually later get converted to estradiol by the action of 17 beta hydroxysteroid dehydrogenases. So either because of the action of aromatase or action of both STS and 17 beta HSD, there may be high local concentrations of the hormone in the cervical tumors. Next, please. So just a little bit about estrogen metabolism, because this is relevant again. So there are 3 different forms of endogenous estrogen found in a woman during the entire lifespan of the woman. Estradiol is the main form in menstruating women. Aromatization of testosterone happens in the ovaries, adipose tissue, and adrenals, and Peyer's patches. Estrone E1 occurs because aromatization of androstenedione and is the chief form in postmenopausal women. E3 predominates during pregnancy. Now, E1 and E2, that is both estradiol and estrone, they get metabolized in the liver, get conjugated into glucuronides and sulfates, and get excreted in the bile. However, gut bacteria, they have the capacity to deconjugate these glucuronides and release estrogen and estrone back into the circulation. So the respective glucuronides, that is E2 glucuronides and E1 glucuronides, are acted upon by the gut bacteria, and E2 and E1 are released back into the circulation. This is known as enterohepatic circulation. Next slide, please. So now, what is estrobolome? Now, gut is, as I've just told you, because Peyer's patches also produce estradiol. So gut is a reservoir of total estrogens. Now, estrobolome is basically the collective enteric bacterial genes which are capable of metabolizing estrogens. So they regulate enterohepatic circulation of estrogens, influence plasma hormone levels, and so this partly determines the lifetime load of estrogen exposure in a woman. Now, another point which is relevant again, which is worth remembering, is that tryptophan non-metabolizing gut flora, like microbiologists in the audience will know that Klebsiella pneumoniae, for example. So these gut flora, they don't utilize tryptophan, and so they let tryptophan be used by the cells of the gastrointestinal tract. They use it by endogenously, and tryptophan metabolism yields melatonin by the cells of the gastrointestinal tract. This melatonin, which most of us know as it's an important hormone which is responsible for circadian rhythm. In addition, melatonin is also anti-proliferative, immunomodulatory, anti-inflammatory, antioxidant, vasoregulatory, and oncostatic. So melatonin acts like a SERM, that is selective estrogen receptor modulator. It prevents the action of estrogen on the cells, and it is also known as a SEAM, that is selective estrogen enzyme modulator, because it modulates the synthesis of estrogen by inhibiting 17-beta HSD1 and sulfatases and promoting the synthesis of the inactive form of estrone, that is E1S. Next, please. So now this, I've just mentioned this, I'm again putting it so that you can just get yourself familiarized with how estrogen is developed and how estrone is again synthesized, and E1S. Next, please. So this is again a cartoon representing what I've just told you. Estrobolome comprises of genes which metabolize estrogen, and you have beta glucuronidases, glucosidases, and sulfatases which release free estrogen in the gut. This promotes release of free estrogen in the gut, and tryptophan non-metabolizing gut flora antagonizes this process by leading to melatonin synthesis, which acts as a SERM and a SEAM and promotes development or synthesis of estrone sulfate. Next slide, please. So this, as a prologue, I think should be more than enough. Now, after this, next step is, so now since we knew that cervical tumors were rich in the hormone estradiol, we wanted to localize estradiol and aromatase in the tissue sections of cervical cancer. And we were fortunate that around the same time, that we were doing this work, IHC antibodies to the hormone was just available commercially. So we used the antibodies to E2 and stained the tissue sections. And you can see this is the part of the tumor, whereas this is the stroma. So we could see, you can see here that the malignant keratinocytes, E2 staining was found intracytoplasmic. See, all this is the cytoplasm. This nucleus is not stained. So E2, what does it do? It synergizes with HPV, stimulates the expression of HPV oncoproteins E6 and E7, induces multiplication and suppresses apoptosis of cancer cells. And in the infiltrate, in contrast to the tumor, you can see that the staining was both nuclear as well as cytoplasmic. So distribution of aromatase was similar to that of the hormone. That is, it was found, aromatase secretion was found both in the tumor cells and also in some cells of the stroma. Next slide, please. Next slide, please. So next later, we wanted to look for whether, cervical cancers express estrogen receptor alpha. So the surprising part was that I just spoke about the K14, HPV 16, E6, E7 animal model of cervical carcinogenesis, where the expression of estrogen receptor was found throughout the evolution of the disease. Whereas in our cases, that is in human cervical cancer, epithelial cells lose ER alpha expression as the disease evolves from normal through precancer to invasive disease. See, this is the slide which is stained for ER alpha, and you can see this is the tumor, whereas this is the stroma. So you can see that the tumor is totally negative, does not express ER alpha, whereas the stroma has cells which look like round cells, which may be lymphocytes, and there are these cells which may be which we thought were fibroblasts. So, and in precancer and invasive tumors, about 30 to 50% of the stromal cells express ER alpha, and expression of ER alpha in the stroma remains throughout the disease evolution. So E2 in the TME, in the tumor microenvironment, E2 is utilized both by ER alpha negative tumor cells by the non-genomic pathway, and by the cells in the TME, they use E2 both via the genomic and the non-genomic pathway. Next slide, please. So as a next step, since we did find some round cells which were using, which had stained positive for ER alpha, we wanted to, and there was in the literature, there was enough information which said that regulatory T cells and the association between regulatory T cells and ER alpha. So what we did was we isolated various populations of cytotoxic T cells, that is cytotoxic T cells, CD8 positive cytotoxic T cells, CD4 positive effector T cells, and CD4 positive regulatory T cells, CD4 positive naive T cells from the peripheral blood. These three subsets were isolated from the cervical tumors. And what we found, that the concentration of estradiol in Tregs, regulatory T cells, was 2 to 3-fold higher when compared to the other subsets. Secondly, we wanted to know whether estradiol in Tregs was complex to, was in combination, was complexed with estrogen receptor. So what we did was we took antibodies to estradiol as for capturing the bound molecule, and then we probed it with anti-ER, and then we found that at least partly estradiol is complexed with estrogen receptor alpha. Next slide, please. So we wanted to check whether a SERD, that is fulvestrant, ICI, whether it will have any effect on the expression of ER alpha in Tregs. So what we did was we used various concentrations of ICI, and we found that with the smallest concentration of ICI also, estrogen receptor expression was almost totally lost. Next slide, please. Next, we wanted to see what this SERD, that is what ICI does to the expression of FOXP3, because FOXP3 is a marker of regulatory T cells, and it is also a master regulator of regulatory T cells. That is, it is a main gene or main protein which controls the activity of regulatory T cells. So we saw that ICI, when it was added to Tregs, not only was ER alpha mRNA lost, even FOXP3 mRNA was lost. There were two types of SERDs which we used, ICI, which is fulvestrant, and another molecule called RU. Both of them were equally capable of destroying, abolishing both ER alpha and FOXP3 expression, both in terms of RNA and protein. So ER alpha protein and FOXP3 protein was also lost. Next slide, please. Next, we wanted to look at the function of how the function of Tregs is affected by the use of ICI or fulvestrant. So we saw that Treg cytokines TGF-beta, there was a drop, 10-fold drop in TGF-beta concentrations and 4-fold drop in IL-10 concentrations, and simultaneously 7-fold increase in effector cytokines gamma interferon and effector cytokine IL-4 in co-cultures which were exposed to ICI. Next slide, please. Simultaneously, the proliferation of effector populations in presence of ICI in co-cultures was regained to normalcy. So that means that ICI was capable of, gaining, of suppressing Tregs and helping effector populations to multiply to the same extent as it does when it is incubated alone. Next slide, please. So in summary, estradiol was significantly increased in cervical cancers and tumor infiltrating regulatory T cells. Blocking ER alpha with a SERD ICI abolished ER expression in Tregs and FOXP3 expression and impaired Treg function. Hence, E2 mediated ER alpha signaling is critical for the sustenance of FOXP3 expression and Treg function via the direct interaction of ER alpha with the FOXP3 promoter. Next slide, please. So this paper was published in Scientific Reports in December 2017. Next slide, please. So we also wanted to see whether estradiol has any action on cervical cancer, cancer-associated fibroblasts. Next slide, please. So what we did was we carried out explant cultures of these cervical tumors, which were freshly dissected, both from early tumors, that is FIGO stage 1B2, and also from late disease, for which we had taken biopsies from patients with cervical cancer, FIGO stage 3A. And we grew them in culture, and then you see these are the cultures, these are the fibroblasts in culture. And since we had, I had mentioned earlier, since we had a clue that some of these cells looked like fibroblasts and ER alpha expression, you can see them here, that it is absent in the tumor and present in the stromal cells. So this gave us a clue that we may be coming across or we may be seeing cancer-associated fibroblasts, at least some type of CAFs, to be ER alpha positive. So after explant culture, I wish to thank Dr. Annapurni Rangarajan here. She's a faculty at IISc. She helped my student, Dr. Mahesh, to to gain expertise for culturing these CAFs for by explant culture. And we saw that these, when we did reverse transcriptase PCR, all the calves had ER alpha. Next slide, please. So we later did this is a heat map showing you the gene expression profiling of cervical cancer associated fibroblasts treated with E2, which was a control, and ER antagonist, MPP, which is a SORM, and ICI, which is a SERD. And we saw that both SERM and SERD modulated genes associated with cell cycle and metabolism, affecting angiogenesis and cancer progression. However, the modus operandi was through different pathways. Next slide, please. So this paper was published in Tumor Biology in the year 2016. Next slide, please. So once again, I wish to just mention that estrogen potentiates HPV-mediated cervical carcinogenesis. How it by a direct carcinogenic action of E2 on cervical cancer tumor cells. This is an early disease, maybe in a pre-malignant state, when the cells express ER alpha. Indirectly, E2 also induces anti-inflammatory and regulatory immune responses. And since E2 is a powerful immunosuppressor, it recruits myeloid-derived suppressor cells from the bone marrow and spleen, and into the from the bone marrow into spleen and tumor beds. It augments estrogen alpha mediated immunosuppression of granulocytic myeloid-derived suppressor cells in cervical cancer. This has been shown by Kozaza et al. In 2019. It promotes VEGF expression. For I'm giving you just one example. Promotes VEGF expression in cervical cancer associated fibroblasts, enabling M2 TAM recruitment. It synergizes with HPV16E7 to increase granzyme B inhibitors, which counters granzyme B mediated killing by NK cells and cytotoxic T cells. E2 via ER alpha controls FOXP3 expression and also Treg function. So, and it also acts non-genomically. E2/3 ER alpha controls CAF function both by the genomic and the non-genomic pathway. So through ER alpha, through ER alpha, it CAF function is controlled genomically. Also, non-genomic pathways are used by the hormone to control CAF function. Next slide, please. So these are two of my last slides. I thought I would just put up some thoughts which can act as food for thoughts for you. So a large population-based breast cancer study using long-term anti-estrogen, especially aromatase inhibitors like letrozole, they saw that this reduced the incidence of cervical neoplasia. This was published by Hess et al. In 2017. So this clearly indicates that even in the clinic, the concept of controlling production of estrogen locally may work. However, you have to keep in mind that STS and 17 beta hydroxysteroid dehydrogenase, both these also have to be inhibited, since nowadays double inhibitors are available for clinical use. And but you first have to, as a first step, document the presence of these enzymes in cervical cancer. Of course, HeLa cell, which is a cervical cancer cell line, expresses both these enzymes. So in most likely you will find these enzymes in the cervical tumor tissues of human origin also. So these two are one aspect which I think holds a better chance of controlling locally available estradiol. Alternatively, repurposing SERDs like ICI, which is fulvestrant, which is already used for treatment of breast cancers, for countering ER alpha expressing pro-tumorigenic cells in the cervical cancer tumor microenvironment, like myeloid-derived suppressor cells, Tregs, and arrest CAF growth also could work, but you have to think of what you will do for the non-genomic action of estradiol, which SERDs will not be able to do anything about. Estrobolums in cervical cancer has not been investigated so far. And a surplus of estrogens in the circulation and or tissue is a risk factor for cervical carcinogenesis too. Now, plasma and tissue estrone levels in patients with cervical cancer is also worth investigating, considering that even is the major form of estrogen in postmenopausal women. Next slide, please. Now, there's a recent study which shows by IHC, they have detected in 65 to 73% of cervical tumors, progesterone receptors have been found. And we all know this is an old story that progesterone has always antagonizes action of estrogen. Now, animal experiments have shown that progesterone has the ability to prevent progression of sin and regresses cervical cancer only when progesterone receptor is expressed and E2 levels are low. Now, this is an animal model where you can't totally extrapolate it to humans. Moreover, this is going to be a rider. E2 levels should be low, and you have just seen my results of our study where we found that E2 levels locally produced are quite high in cervical tumors. So I'm not too sure how progesterone administering progesterone in cervical cancer, how well it will work clinically. Next slide, please. So I have a very pleasant duty to perform. I wish to thank Dr. A.J. Rao and Annapurni Rangarajan of IISc for having helped us at crucial moments. I wish to thank my graduate students, late Dr. Srinivas Adurti, Dr. Mahesh, and junior research fellows Advait, Alka, and Vinod. I also wish to thank my collaborators, Dr. Bafna, Dr. Uma, Dr. Geetashree Mukherjee, and Dr. Acharya, and Department of Biotechnology, ICMR, and CSIR for funding for both my projects as well as my students during the journey. I thank you very much for your patient hearing and would be happy to answer questions. Thank you.
Thank you so much, madam. That was really a path-breaking experiments that you have shown us, and then you demonstrated very clearly that in pre-invasive and early invasive, yes, estrogen is very, very carcinogenic, and you have demonstrated beautifully with all those specimens and markers that you had shown. So what about the ones which are recurrent tumors? I mean, madam, it's the same scenario even there, or because we know the tumor is always growing inside in post-treatment or during the process. So the levels should be the same or more estrogen levels locally?
So we haven't done that work, but anyway, I guess, I mean, if I'm allowed to guess, I think it should be there, because for two reasons. One is that local production of estrogen is happening. Secondly, Tregs, we have done Treg sampling even from males, male donors. So blood donors.
I've seen that, madam. That male donors, I was also surprised how it was.
Right. So blood donors from males, these also had high levels of Tregs, which means that, even the lymph nodes have adipose tissue. And so that acts as a local supply of estrogen. So in fact, I've not mentioned this, but we did some, IHC, because I was so excited when I came across our initial findings about Tregs that I wanted to see what the scenario was in other solid tumors. So we've sampled, we have done IHC, we've not published this, but I'm just mentioning here. We've done IHC for estradiol and estrogen receptor from various male solid tumors taken from males. And lo and behold, it was loaded with estrogen and estrogen receptors. So this is something which, it's very interesting. And moreover, since there are ways of checking these, it is worth taking it up as independent studies. If whoever's interested, it'd be really useful.
Thank you so much, madam. It was very eye-opening for us to know so much is involved in these cancer patients. And yes, all of these cancers do occur, or at least the infection would have started during the reproductive age group only, and which we see postmenopausal or during the perimenopausal age. So surely there might be a role, and because the cervix also undergoes so much of changes because of hormones, again because of estrogens.
Yes.
So maybe like in future, we may see some things related to this. Thank you so much, madam.
My pleasure. Thank you very much.
Thank you. So now we move on to the next topic for the day, that is immunotherapy in recurrent cervical cancer. This talk is being given by our medical oncologist, senior medical oncologist, Dr. Ravi Diwakar. Sir is a gold medalist from Adyar Cancer Institute, and he's done his medicine from Kasturba Medical College, and he's been a senior consultant in medical oncology in Bangalore for the last 25 years. So sir has vast experience, and he's published many national and international papers and articles. So here we have Dr. Ravi Diwakar.
Keyboard. Keyboard.
Thank you, Dr. Annapurna. From a lot of basic science involving carcinoma cervix to a clinical topic. I will be talking today about immunotherapy in recurrent metastatic cervical carcinoma. We all know that metastatic disease will develop in a significant population of patients with cervical cancer, and usually it happens within the first couple of years after completing treatment. And in majority of these cases, this metastatic disease is not curable. There are a small subgroup of patients who may present with metastatic recurrent disease, which is loco-regional or with limited or oligometastatic disease, where surgical treatment is a possibility. And these patients may be potentially curable with surgery, or the patients may even have a long-term survival with chemotherapy. We have seen patients who come with isolated pleural effusion or a supraclavicular node metastasis or a paralytic lymph node metastasis, which cannot be operated for various reasons. We have given chemotherapy, they have survived beyond 5 years also, but they constitute a small group of patients. In a study, we found there was an estimated prevalence of organ site involvement, like pelvic or paralytic nodes in 75 to 62%, lung, liver, peritoneum, adrenal, intestines, and even the skin was found to have recurrent or metastatic disease. And majority of these patients with metastatic disease who are not candidates for local treatment will receive chemotherapy. And today, the first line setting for chemotherapy, first line treatment would be a chemotherapy plus bevacizumab rather than chemotherapy alone. Because in the GOG trial, it showed that incorporating bevacizumab improved the overall survival in this group of patients. And this is what the guidelines said in 2019, 18 and all NCCN guidelines. The first line treatment was paclitaxel cisplatin with bevacizumab or carboplatin, or you could use topotecan for some reason where you could not use platinums. Only chemotherapy was lower in the pecking order. And this was the trial which said bevacizumab showed overall survival, where 452 women with metastatic persistent or recurrent cervical carcinoma were randomized assigned to chemotherapy alone or chemotherapy with bevacizumab. Now, there was a significant improvement in overall survival compared to chemotherapy alone with a hazard ratio of 0.77. And if you took patients who had not received radiation earlier for their earlier disease, the overall survival was still better in both the groups, whether they received bevacizumab with or without bevacizumab. And there was a significant improvement in the overall response rates also. And this is what led to the standard of care which was there presently. Now, from here, where to go? The next option was to go to immunotherapy. Now, there are a lot of things which are there in immunotherapy, which in cervical cancer, but we will concentrate on probably immune checkpoint inhibitors, because these are the in thing now in medical oncology. And basically, the checkpoint inhibitors include antibodies targeting CTL4 or PD1 and PDL1. I will just briefly cover to what this checkpoints are and what is this thing. Now, the rationale for immunotherapy in cervical cancer is the gynecological tumors are known to evade the immune system by inhibiting antigen presentation, recruiting immunosuppressive cells like the Tregs, and secreting immunosuppressive factors like IL-10 and all those things. The other thing is cancer. The cervical cancer is associated is a viral associated disease, and it is known to have high mutational burden, which could say that, which could indicate that the immune checkpoint inhibitors may be effective. Now, basically, to say how the immune checkpoint, what are the immune checkpoints and how they work. Now, you have the antigen presenting cell. This presents the antigen on the MHC complex, and it is recognized by the T cells. Once this happens, along with the concerted co-stimulation of CD28B7, there is a positive signal. The weak TCR signal becomes a positive signal, and this leads to secretion of IL-2 proliferation and increased survival. So what happens is, when the TCR signal becomes positive or strong, there is a negative feedback. You have the CTL4 antigens which are there in the stored intracellularly. They are pushed extracellular to the surface, and this CTL4 competes with CD28 for the B7 molecules. And when it binds the B7 molecule, there is reduced IL-2 production, reduced proliferation, and reduced survival. Probably this is a protective mechanism in our body to prevent over-exaggeration of the exaggeration of the immune response to whatever antigen that is presented, or for protection against autoimmune diseases also. Then this is the other pathway. Then what happens is, when the antigen is bound to the T cell receptors, the T cell receptors become activated and they produce cytokines like interferon, IL-2, and this leads to activation of the T cells, and hence this thing. At the same time, to control this immune response, there is upregulation of the PD1, which is the programmed death cell receptor on the T cells, and PDL1s. This PDL1 is present in the tumor cells. And when the PDL1 and PD1 PDL1 binds to the PD1, there is reduced proliferation, reduced cytokine production, and reduced survival. So this is just a schematic representation of the same thing again. How, when blockade happens, what happens when there is a CDL? If you block the CTL4 receptor by an anti-CTL4 antibody, it allows activation and proliferation of the T cell clones and reduces Treg mediated immunosuppression. And because of this, again, along with that, if you block the PD1 pathway by an antibody to the PD1, that is anti-PD1 or PD1 or an anti-PDL1 antibody, what happens is it restores the activity of the quiescent T cell. And this dual pathway, that is blockade of CTLA4 and blockade of PD1, could have a synergistic effect and resulting in larger and longer lasting anti-tumor responses. Now we have various antibodies that are available in clinical practice. We have the CTLA4 inhibitors like ipilimumab and tremelimumab, and PD1 inhibitors like pembrolizumab, nivolumab, and PDL1 inhibitors like atezolizumab, avelumab, and durvalumab, and they are in use in several solid tumors as well as hematological malignancies. Now, how do we select which patients would respond to a checkpoint inhibitor or not? Now, this is based on the certain receptors. Now, you have the PD-L1, like PD-1 and PD-L1, which can be assessed on the cell surface by an immunohistochemistry test. And the other thing is you look for microsatellite instability or mismatch repair deficiency. This is important because it is known that MSI high tumors or tumors which have a compromised DNA repair ability have more genomic instability, and it may make them more susceptible to immunotherapy. And the third one is, I already said, tumor mutation burden. This measures the number of mutations in a tumor. This microsatellite instability is assessed by next generation sequencing, but the same thing can be done by IHC by looking at the MMR deficiency deficient genes by IHC. The tumor mutation burden is detected by using a chromosome genome profiling. And here what happens is the mutations in the cancer cells result in modified surface proteins and lead to production of neoantigens. And these neoantigens are thought to be targeted by the immune system. So higher the tumor mutational burden, higher may be the presence of neoantigens on the cell surface, and higher may be the chances that they will respond to immunotherapy. And I already showed you that CS carcinoma cervix has a tendency to have high tumor mutation burden. How do you detect these things? So I already told you the DMMR proteins can be detected by IHC, and TMB can be detected if there is a high tumor mutational burden. You can look into it by doing a comprehensive genome profiling. Whereas for the immune checkpoint inhibitors, that PD-1 or PD-L1, what is this thing is you do the IHC by using this antigens like C28A8 or a 22C3, and you do the scoring on the cells after IHC. And for cervix, what is important is to do the CPS score or the combined positive score. And if the combined positive score is more than 1, then these patients can be eligible for using immunotherapy drugs, and that is what is depicted. A CPS score is used in cervical cancers. Now, this is basically a graphic interpretation of the same CPS score. In CPS score, what you do is you see the number of cells, the tumor cells, lymphocytes, and macrophages, which have the PDL staining, and you divide that by the total number of tumor cells and multiply by 100. That will give you the CPS score. That is called as the combined positive score. So after all this, they were coming to the clinical trials that showed how immunotherapy could be effective in cervix. One of the initial trials was a Checkmate 358, which used nivolumab in recurrent or metastatic cervical cancers. In this trial, it included a lot of other vulval and vaginal cancers also, but there were about 19 patients with cervical cancer. In that, about 26% of these patients showed an overall response rate, and the duration of response ranged from 23 to 29 months. This was the Keynote 158 trial where they used pembrolizumab as a single agent in previously treated advanced cervical cancer. There were 82 patients, and they had a median duration of response ranging from 3.7 to 18.6 months. And this Keynote trial, irrespective of the PD-L1 positive or this thing, it showed a response. There were CRs, a partial response, and standard stable disease accounting for this thing. And in the PD-L1 patients, it was the disease control rate was 36% here and about 31% here. About 12% experienced treatment-related side effects, and immune-related adverse events were seen in about 25%. Based on these findings, the FDA approved gave an accelerated approval for use of this drug in patients who were PD-L1 positive with a CPS score of more than 1% in advanced cervical carcinoma after progression of chemotherapy or the first-line therapy of chemotherapy with bevacizumab. Then came the trial where pembrolizumab was used as first line in persistent recurrent or metastatic cervical carcinoma, the Keynote 826 trial. This was the eligibility criteria. They said no prior systemic therapy should be used, but if they had received radiotherapy or chemoradiotherapy in the initial phase of their treatment, it could be acceptable. They stratified according to the presence or absence of metastatic disease at diagnosis. PD-L1 CPS less than 1 versus 1 to 10 and more than 10. And if there was bevacizumab also was used, and if they had used bevacizumab or not. Bevacizumab use was acceptable. So pembrolizumab was given 200 milligrams IV every 3 weeks for up to 35 cycles, that is up to 2 years, along with paclitaxel up to 6 years, and bevacizumab 15 mg per meter square every 3 weeks, as long as the patient wished to take. And the primary endpoints were overall survival and progression-free survival. And you had secondary endpoints of overall response rate, duration of response, and safety. This was the schema. 883 patients were screened, 617 underwent randomization. 308 were assigned to the pembrolizumab, and 309 were assigned to the placebo group. And as on date, 104 continue to receive treatment in the pembrolizumab with at least one drug, whereas 54 patients in the placebo group continue to receive at least one agent. Now, these were the characteristics of the patients. They were well matched in for age, sex, the ethnicity, the ECOG performance status. Only patients with ECOG performance 1, 0 or 1 were used. Beyond 1, patients were not considered for the clinical trial. And stages also matched almost equally between the two groups. Then the PD-L1 scoring and previous treatment, use of bevacizumab, all those things matched equally. The bevacizumab, 63% patients in the pembrolizumab group used, 63% used bevacizumab, 36% did not use. There it was 62 and 37. Probably only the bias was with respect to the histology type. In this trial, they did not stratify according to the histology. And in the pembrolizumab group, you had a more favorable histology, probably because the squamous cell carcinomas were 76% and adenocarcinomas were 18%, whereas in the placebo group, we had 68% and 27%. This was probably only bias in the patient population. Now, if you look at the survivals, like progression-free survival, if you look at when the PDL score of CPS more than 1, there was a improvement with a hazard ratio of 0.62 with addition of pembrolizumab. When you looked at progression-free survival beyond 10%, when the CPS score was more than 10%, the hazard ratio was 0.58. And in the entire intent to treat population, the curve still separated with a hazard ratio of 0.65. And if you look at the forest plot, you had responses favoring pembrolizumab, the progression-free survival favoring pembrolizumab, except for the PDL score less than 1. So if the PDL score was more than 1 and in any other subgroup that was analyzed, it showed a tendency to progression-free survival. Now, looking at the overall survival, this also presented in the same way. If you had an overall survival in the PDL score of more than 1, favored pembrolizumab with a hazard ratio of 0.64. In patients with more than 10, the hazard ratio was 0.61. And in the entire intent to treatment group, it was 0.61. And if you looked at the forest plot, the same thing. It favored use of pembrolizumab in all the groups that were studied, except probably in the PDL1 CPS score less than 1%. And if you looked at the adverse events, most of the adverse events were because of the chemotherapy and placebo. And there were almost everybody had some adverse event. The main adverse events were hematologic, like anemia, thrombocytopenia, and this thing, and peripheral neuropathy. These were this thing, and they were comparable in the both the pembrolizumab and in the this thing. So pembrolizumab did not add to the toxicity of chemotherapy or bevacizumab. If you look at the immune-related toxicity, again, you had about some amount of about 33-35% of immune-related toxicity in the pembrolizumab group, but most of them were grade 1, grade 2. Very few people had grade 3, grade 4, and most of them were treatable, like you had thyroid-related problems, colitis leading to diarrhea, skin-related problems and all. Major organ dysfunctions like hepatitis, pancreatitis, pneumonitis, myocarditis and all were very minimal. So most of the immune-related adverse events are also very tolerable and treatable. So based on this, the conclusions were there is a first immunotherapy study to incorporate PD-L1 inhibitor along with chemotherapy with or without bevacizumab. And addition of bevacizumab is optional because even without bevacizumab and only chemotherapy and PD-L1, this thing you had survival. Survival was essentially the same. And the overall survival benefit was 24 months. At 24 months was 53% in the pembrolizumab and 41% in the placebo arm. And this was about 12% difference. And PFS and OS benefits were persistent in the protocol specified subgroups with a CPS score of 1 or more. Probably the only critique of this study I could think of was the pembrolizumab contained a more favorable histology group with more squamous cell carcinoma and less adenocarcinoma compared to the placebo group. So this led to the NCCN guideline of 2022 putting this combination as the preferred, one of the first preferred regimens, and either with cisplatin or carboplatin, and use bevacizumab and this thing lower in the order. But if you look, if you have used pembrolizumab, if you had not used pembrolizumab, pembrolizumab could be used as a single agent in the second line. But if you have already used pembrolizumab and patient progresses, what was there? What was there was only the chemotherapy drugs. And in these drugs, with these drugs, the response rates were hardly 10 to 15%. But this is one drug which has come up now called tisotumab vedotin, which is an antibody drug conjugate, which is used against a tissue factor. It's a protein which is expressed in several solid tumors, including carcinoma cervix. Now, what is an antibody drug conjugate? Is you have a monoclonal, you have an antibody, which is the IgG antibody, and to that you attach a cytotoxin that is like a chemotherapy drug, and that is attached to the help with the help of a linker. Now, what this does is when you inject an antibody drug conjugate into the patient, the antibody drug conjugate binds to the tissue factor, which is present on the surface of the cell. And once that happens, the antibody drug conjugate and receptor complex is internalized. And once it is internalized, the lysosome degrades it and releases the cytotoxin. And this cytotoxin is a chemotherapy drug which disrupts the microtubules in the body and leads to DNA strand breakage. And because of this, it causes cell death, including apoptotic cell death and cytotoxic cell death. And the drug that is used here is a MMAE, that is monomethyl orostatin E. That is the drug. It's a microtubule disrupting agent. In addition to that, binding of the antibody drug conjugate to the receptor or the tissue factor receptor on the cell surface induces immunogenic cell death, activates antibody dependent cell mediated cytotoxicity and antibody dependent cellular phagocytosis, and produces its bystander effect also. Now, this drug conjugate was studied in a phase 1-2 trial called Innova-TV 201 study, where patients with relapsed, advanced, or metastatic solid tumors were all solid tumors were treated, of which there were 34 patients with cervical cancer. And in these 34 patients, 9 patients achieved an objective response. And there was grade 3 toxicity about 63%, and major toxicity was seen in the ocular, along with the other usual toxicities that you see with antibody drug conjugates like thrombocytopenia, neuropathy. In addition to that, they saw some ocular related toxicities. Now, there are 2 ongoing studies with tisotumab vedotin in cervical cancer. One is called the NGOT CX12 study, where it is a phase 3 study seeing tisotumab vedotin was the investigator's choice chemotherapy in recurrent cervical cancer, first line. And you have the Innova-TV 204 study. It's a phase 2 study where tisotumab vedotin is tested in previously treated recurrent or metastatic cancer. And the results of these trials are out now. This was presented in Lancet Oncology. And the study period was from June 2018 to April 2009. The data cutoff was at February 2020, median follow-up of 10 months. And in this study, 4 patients are still on treatment and 33 patients are on follow-up. There were a total of 101 patients. And the median duration of treatment was 4.2 months. And the median number of doses received was 6. Now, summary of the response by an independent review committee. Of the 101 patients, there was an objective response rate in 24%. That included 7 complete response and 17 partial response. If you take stable disease, there were about 49% stable disease. So total disease control rate was seen in 72%. This is in a patient who had already received chemotherapy. And duration of response was 8.3%. And time to response was quite short. It is 1.4 months. That is roughly in about 2 cycles of treatment, you could see a response. And if you look at the 6-month overall survival, it was 79%, and the 12-month overall survival was 51%, which is quite major change that we see in pre-treated patients. This is a waterfall plot showing the same thing in different patients. And this is the swimmer's plot also showing the same thing. So there are quite a few patients who have stopped the treatment. Like this purple thing indicates that it is off treatment, and they still have an ongoing response. And this is a schematic response again. If you look at the 12-month period, about 50% are alive with surviving this post the treatment with this. If you look at the toxicity, as I told you, there are classical toxicities of the chemotherapy drug like alopecia, thrombocytopenia, neutropenia, neuropathy, and all. But hardly any grade 3, 4 toxicity. Most of the toxicities were grade 1 and grade 2. And specific to the drug was conjunctivitis, dry eye, keratitis, and those sort of problems, eye-related problems. So which requires that if you should take a proper ocular opinion and consultation and be in touch with an ophthalmologist during the course of this treatment. Now, what is there in store in the future? What other trials are there? You have one trial called the BEAT-CC trial, which is a phase 3 trial where they have used atezolizumab with chemotherapy and bevacizumab. This is a PD-L1 inhibitor. Now there are trials where they are using pembrolizumab with chemotherapy versus chemotherapy in locally advanced cervical carcinoma. And you have durvalumab, which is a PD-L1 inhibitor used with or without chemoradiotherapy in locally advanced cervical cancer. You have another new drug called bastilimab, which is a PD-1 inhibitor, and zalifrelimab, which is a CTL4A inhibitor. That combination, with or without combination, is being used in 2 phase 2 clinical trials. And you have a bifunctional fusion protein, fusion antibody, or a biclonal antibody conjugate, where you have a antibody called binatrafus alpha, which blocks both PD-L1 and TGF-beta. So they are bifunctional monoclonal antibodies, and these are all in clinical trials. One other approach, immunotherapy approach to cervical cancer, is to use TILs, that is tumor infiltrating lymphocytes. It has been known that higher TILs associated with more favorable prognosis in cervical cancer. So what you do is, when you resect the tumor, you take fragments of the tumor and isolate the TILs. You expand the TILs by in ex vivo by to the power of 10 to the power of 9 to 10 to the power of 11 cells, and do then the lymphodepletion and infuse these TILs along with IL-2 into the body. Now, with this sort of TIL immunotherapy, there was a trial where in the initial report said there was an overall response rate in about 44% of patients. And some of the patients even showed a CR, PR, and stable disease. Majority of the patients showed some response, and CR was seen in about 3 patients, PR in seen in about 7 to 8 patients, and stable disease in about 4 patients. So, but my take is, all these treatment options come at a high cost with lot of side effects. It's far more easier and economical to vaccinate prepubertal children against HPV and prevent the disease. Whenever we talk about HPV vaccination to children, the issue that comes up is cost. But if you want to do the first line therapy of pembrolizumab with chemotherapy plus or minus bevacizumab for 2 years in these patients to respond, you would be spending 1.5 to 1.6 crores. I don't think it is worthwhile. So I think prevention is always better than cure. Thank you.
Thank you so much, sir, for summarizing all the immunotherapy trials and the mechanism as to how the immunotherapy is acting in cervical cancer. Sir, can you just summarize what are the common side effects that you see in clinics, that we see in clinic day-to-day clinics?
With immunotherapy drugs?
Yes, sir.
Yeah. See, common immunotherapy side effects that we see is hypothyroidism, some skin-related toxicities, skin reactions could happen, and colitis leading to diarrhea. And most of these are manageable. You don't have to reduce the dose or delay the dose. You use symptomatic treatment. Most of the grade 1, 2 toxicities can be treated with symptomatic treatment. If it is grade 3 or beyond that, you use steroids for these patients, and most often they recover. If patients have prolonged side effects like these side effects, then you hold the treatment till the patient recovers to a grade 1, grade 2 and restart the treatment. Very rarely have we seen patients where we had to stop treatment. I have no experience of using pembrolizumab in cervical cancer, but we have used pembrolizumab in other solid tumors, and this is what we see. We have rarely seen a patient who has developed uncontrolled blood sugars, pneumonitis or hepatitis, or those sort of problems. Hardly experience those sort of problems. Most of the drugs, most of the side effects can be manageable. Now, there is thinking that there is some difference in the side effects between PD-1 and PD-L1 inhibitors. What we use is the pembrolizumab, which is a PD-1 inhibitor, where the side effects are less. The side effects, the immune-related side effects, may be more with the PD-L1 inhibitors like atezolizumab or durvalumab is what is the thinking. Most of the times we have we can manage this side effect effectively without hindering the treatment option.
Thank you so much, sir.
Thank you, Dr. Annapurna.
Thank you, sir. Now, shall we go on to the last topic of the day? It is by Dr. Priya Kyrgiou from Imperial College London, and Madam is going to talk on vaginal microbiome and cervical cancer. As we have seen the last, may I have Madam CV? Dr. Maria, hi. I will just introduce you first to the audience. I will just show your CV. After that, you can go online with your talk. Thank you so much, Dr. Maria, for joining. And today we are all looking forward to this topic of vaginal microbiome and cervical cancer. So, as you all know, Professor Priya Kyrgiou is a professor and consultant gynecological oncologic surgeon, and she is the head of section of gynecologic oncology. And she also is IRDB Department of Gut and Metabolism and Reproduction, Surgery and Cancer of Imperial College London. She works at West London Gynecological Cancer Centre, Imperial Healthcare, NHS Trust. Next, please. Can you move the slide? Her main clinical interest is in gynecologic and gynecologic cancer surgery and minimal access surgery. And the primary focus of her research is the translational research in gynecological diseases and cancers, in particular cervical, endometrial, and ovarian cancers, with an emphasis on prevention and personalized treatment. She has a deep interest in cervical pre-invasive and invasive diseases, and primary cervical cancer screening and HPV vaccines. Next, please. Madam had led research into the impact of treatment of cervical cancer, pre-invasive and early invasive cancer, cervical diseases, and the reproductive outcomes, and the clinical use of HPV biomarkers in personalizing the management of women with abnormal findings at primary population-based screening. Madam runs a research group investigating the impact of obesity and metabolic disorders in the development of gynecologic cancers, with an emphasis on endometrial cancers. So, Madam also investigates the role of genital tract and the gut microbiome and host interactions in the development and treatment response of gynecologic cancers. Madam has 205 publications, and including the Cochrane reviews. So here we have today with us Dr. Maria Kyrgiou from Imperial College London.
Can you see my screen?
Yes, ma'am.
Great. Let me just go back. Well, it's a great pleasure to be here, and I want to thank you all for this kind invitation. I heard some interesting presentations, and we're going to move in the last, I think, presentation of today's meeting on the genital tract microbiome and gynecological cancers, with an emphasis on HPV and cervical cancer. And I think we all agree that it is an area that has become of particular interest to the literature over the last 10 to 15 years. It all sort of started when the NIH initiative launched the Human Microbiome Project in 2007, with the awareness that for every 1 trillion of human cells, there are 10 trillion of bacterial cells on the human body, that our body is heavily colonized by these bacteria, and that these are very important into what happens in the human body, into describing health and disease. And the project by NIH started with the ultimate goal to demonstrate whether there are opportunities by using and understanding the human microbiome to be able to monitor and even manipulate this towards promoting health. Something that we all suspected, but we had limited, let's say, techniques that were able to bring into understanding the full taxonomy on a specific part of the human body. And of course, as with everything that is so novel, there was a huge interest for several years that followed for people that try to look at different cavities in the human body that are particularly dominated by the microbiome. For example, the gut was the first, and is one of the first things that has been investigated. The bacteria that dominate the human gut are those that may be very important in the way our immune system works, in the way that our diet affects our immunity, in the way that our body is able to use this bacteria to metabolize substances or even drugs and food, and how all of this is important in what ultimately happens in our body, and what we suspect about healthy lifestyle and how this translates into increased malignancies in some cases. And of course, for us gynecologists, the cavity that is very important to us is the vagina that is also dominated by bacteria that evolutionary have developed there over millions of years, and are those that are actually very important in maintaining health. And of course, if we look at the female genital tract and the cancers that we as gynecologists are interested in, from the cervix to the endometrium to ovarian cancer, the challenges are different. For the cervix, it's to understand what makes this very common infection by this virus to cause problem in a small proportion of individuals that get this. In endometrial cancer, to see how we can understand what leads to the development, prevent and improve the treatment, and the same for ovarian cancer, particularly with the knowledge that this now initiates from the fallopian tubes in a significant proportion of the cases. And of course, you all remember Albert Dodlein, the person who first described lactobacillus a good bacterium that creates a low pH, produces lactic acid, bacteriocins, and dominates the female vagina, and is very important because it prevents the development of different pathogens.
Excuse
And
me,
of
ma'am.
course,
Dr. Maria,
Yes.
can you please share your screen?
Can you not
had
see the
disabled.
screen?
Yes.
Can you not see it now?
No, ma'am.
No. How about now?
Yes, we can. Thank you.
So you didn't see any of those slides, yeah?
Yes.
Can you see it now?
Yes, yes. Thank you. Thank you.
Let's see, because I'm a bit confused. Can you see the full screen or just the can you see dotted line at the moment? Albert dotted line on the picture.
We can see the whole thing now. Yes, we can.
Yeah?
Yes.
That
we can see.
microbiome, yes, ma'am.
Okay, the picture with lactobacillus, yeah? Because I have two screens and I don't know which one plays on your screen.
The vaginal microbiome, that's what we see, lactobacillus.
Both
Yeah,
are seen,
yeah,
madam. Both are seen. Even that Albert Dodelin also is seen. The whole thing is seen.
Good. So there was a number of pathogens and anaerobes like Gardnerella, Prevotella, Smithia megasphaera, all of this under the umbrella of what we used to call with traditional cultures bacterial vaginosis. And this in its own has been associated over the years with a number of diseases for women, preterm birth, the chances of getting HIV, sexually transmitted diseases, and of course, for what this talk is about, the human papillomavirus. And there have been some meta-analyses and systematic reviews that associated HPV infection and cervical intraepithelial neoplasia with the presence of bacterial vaginosis. And that raised the first hypothesis that actually what happens in the remaining of the bacterial flora within the vagina is important in telling us whether there is the presence, there is an increased risk of getting these infections. And at the same time, in different areas of the genital tract, there have been some indications in big papers like the one in Lancet Oncology in 2011, that women that have a history of pelvic inflammatory disease have a high risk of getting endometrial and ovarian cancer. And this principle that these pro-inflammatory environments may be promoting oncogenesis in some of the organs of the female genital tract. And now particularly that we know loads of high-grade serous cancers originate from the end of the fallopian tube, this history of inflammatory processes in those tubes may be important into carcinogenesis. So of course, what really changed over the last few years is that the hypothesis was there. I just think that we now have the technologies to be able to go beyond the traditional culture techniques, which are not particularly accurate, that tell us only what is there in large amounts, to get a full taxonomy with next generation 16S RNA sequencing that is able to look at a specific gene within bacteria and be able to describe in great length the different genera, the different species that are available within that area. And one of the very known papers by Jack Ravel in 2011 was the one that defined, if you wish, in sexually active premenopausal asymptomatic women with no pathology, the different kind of dominant community state types, as he called them, dominated by lactobacillus in 4 groups, Crispatus, Gasserianus, and Jensenii. And this 4th group of lactobacillus deplete microbiome that has high diversity, within us being that transitional state between those 2 microbiomes. And of course, people looked at how different practices, ethnicity is important. You can see here in the same study how they looked at the microbiome composition according to ethnicity. You can see that Afro-Caribbean women had a 40% of CST4, much higher than Caucasian women, for example, where this is 10%. And this may explain some of practices in those countries of practicing lavage, given the odor that sometimes is produced by these anaerobes. And of course, we understood over the years how different environmental factors, hormones, douching, smoking, menopause, contraceptive pill, and of course, pregnancy may all cause a major shift into the microbiome in women. And of course, the menstrual cycle itself, which happens every month, is also very important in the balance of the microbiome, and how the estrogen, menopause, the availability of glycogen may all be important in the presence of abnormalities of normal or pathology in the microflora. So coming back to HPV, we all know that human papillomavirus is a very, very common virus that pretty much infects the majority of sexually active women. And it's a virus that has found this weak embryological zone within the cervix, the so-called transformation zone, to find a weak area embryologically and cause disease. But we all know that HPV is common, and actually getting cancer is a rare complication of a very, very common infection of the cervix by this virus. And because the cervix is so easily accessible, over the years we were able to sample the cervix frequently and understand what we call the natural history of this disease. And at the same time, over the last few years, we have more techniques in our hands that allow us also to understand better the life cycle of the virus, how this gets access into the cells of the basal membranes, and use the mechanisms of the cells to express the oncogenes within the epithelium. So it's a very complex system that we try to understands what makes clearance and persistence possible in different women, how the E67 oncogene expression, the genomic instability, the viral integration is important, how different environmental factors may play a role, and how in all of this we have the role of the microbiome. And we all quote to our patients when we see them in the clinic this invert pyramid of how many cases can regress, how many cases can progress, how many of these cases go back to normal without any medical intervention. But what we still have not really understood is what makes this pyramid to go towards the one or the other infection. What is the factor that makes two CM3s that look entirely the same under the cytological microscope, in one case to regress back to normal and clear the infection, and in another case to progress into an aggressive invasive cancer and lymphatic metastases? And this is where all of these factors from genetics, epigenetics, metagenomics come into understanding how the biology of the whole system actually works. And we have to bear in mind this because we can't look at each one of these factors individually, but we need to explore each one of these factors individually. The host has its own genetic and environmental factors, its own immune responses, its own risk factors from people's practices like smoking, different kind of epigenetic changes. At the same time, the virus has different subtypes, some of which we know have higher oncogenic potential. We know that epigenetic and methylation changes in the virus itself may alter its oncogenic potential. And of course, in the middle of this, we have this microbiome that sits there, but is probably very, very important into the interactions between the host and the HPV. So, of course, when all of this started about the microbiome, there were many authors that looked into different evidence. So, for example, this small study back in 2013 of 35 pairs of monozygotic twins, i.e. girls that had exactly the same genetic material, looking at the microbiome and the presence of HPV infection, and they found decreased lactobacillus in women with HPV in those monozygotic twins, with Smitha being one of the microbiological markers of HPV infection. And of course, looking at persistence, acquisition, and clearance, having data from Brutman in a small proportion of women that were sampled biweekly, finding how different microbiome may be important in what this, whether the HPV infection will be cleared, but obviously in a very small number of studies. So one of the first papers that we have published back in 2015 was the first one to describe in a cross-sectional nature the actual microbiome according to disease severity. And we found that the more severe the presence of disease, the actual lowest the rates of lactobacillus. So the more significant your precancer is, the lower the rates of dominance of the good bacteria called lactobacillus. The second thing, obviously, that has been a matter of significant discussion is what really this does to the so-called upper genital tract. What happens higher up into the endometrium, in the fallopian tubes in the ovaries? These cavities for several years were thought to be completely sterile. And the question is whether this is actually a true reflection of what happens, or whether we just didn't have culture techniques that were sensitive enough to describe the taxonomy in areas that probably have a low biomass of microbes. For example, for many years, we thought that the endometrial cavity is sterile, but we're beginning now to change the opinion on this. And we know that many cancers are associated with low-grade inflammation, and the belief that the areas have been completely sterile may have not taken into account the importance of this, which is predominantly the case with obesity and metabolic diseases. A few years back, we also did another study where we described the vaginal microbiome in women who were obese and women who were not obese, with a BMI of more and less than 30. And we found increased diversity and increased prevalence of lactobacillus deplete communities in those individuals that were obese with a BMI of more than 30. We found decreased abundance of Lactobacillus crispatus, which is the major one that is described as the so-called healthy microbiome, high local levels of cytokines in those deplete individuals. And when we looked at the subpopulation of women who undergone bariatric surgery with longitudinal sampling for up to 6 months, there was high transition from bacterial diversity and increased dominance of lactobacillus. There have been studies that try to recover bugs from the endometrium. For example, one that used PCR and not next generation sequencing found 95% microbial recovery from the upper genital tract. There was another paper in Nature Communications that found distinct microbial communities in the cervix, the fallopian tubes, the peritoneum, the ovaries, and the uterus along the female genital tract. Of course, this is where new evidence and new research comes into the mix. And I think these papers became very popular, lots of people trying to understand this evidence. But a paper that followed put a lot of question marks as to whether this is actually the case, and started discussing how important contamination is of the samples and how easy it is to call an area non-sterile, although you're just describing contamination that can happen during the sample collection, during the sample processing, even the air, the bench, the swab that you're using, the knife that you cut the sample. All of these are extremely important and can lead to substantial contaminations, which then may give you the impression that there is actually bacteria there when actually there isn't. And this is very important, particularly when you look at what we call low biomass samples, samples that are actually from areas that are not dominated by bacteria. And many of these reports, like the one in Genome Medicine that talk about successful amplification of bacteria from the lower genital tract and upper genital tract, may be actually overinflated because they did not use thorough techniques to remove the contamination of those samples. In the fallopian tubes and the ovaries, things are also very interesting. A lot of high-grade serous cancers originate from the fallopian tube. There is evidence in major journals like Lancet Oncology that if you've had, as we discussed, pelvic inflammatory disease, your risk may be higher. And therefore, it is very important to try to explore the role of the microbiome in those cases. And it's very important to see how we can understand what really happens in those areas, but equally the major challenges into sampling those areas, because they're not as accessible as the cervix or the endometrium, and trying to understand what really happens there is obviously restricted by the difficulty in obtaining samples. But however, some people tried to find and describe in ovarian cancer samples that were operated the presence of microbiome in different sites, again with the difficulty of possible contamination. And then this major study came in Lancet Oncology, looking into the role of the microbiome in ovarian cancer. And they found that ovarian cancer patients were associated with CST0 microbiome, community state type 0, as they called it, that is less than 50% dominated by lactobacillus, particularly in women over the age of 50. And the authors concluded that this may be the only modifiable cell non-autonomous mechanism, and probiotics may affect the female genital microbiome higher up. And this may be something we can do to manipulate it and reduce the incidence. And that was a very sudden and massive paper in an area that has been very little known about. The other very important thing that has come as hypothesis in the literature is to see how different microbiome impacts of the gut microbiome may be a possible modifiable factor that can affect even response to chemotherapy. There's been loads of evidence to see how the gut microbiome structure affects activation of the immune response against the tumor, and the dynamics of this gut microbiome needs to be associated with chemotherapy response in those individuals. In our group, we tried to do some similar kind of work in cancer patients that we operate on a regular basis in our institution. And again, in the context of cross-sectional studies, i.e. studies that just describe associations, and this is the first step in every research, but not sufficient, as we're going to discuss later on. We looked at the microbiome continuum along the female genital tract. And you can see in this bar that we've put a very stringent protocol into ensuring that we don't pick up just contamination in the way the samples were collected, in the way that if a uterus is pulled out of the vagina when you do a laparoscopic hysterectomy, what you're probably picking up in the upper genital tract by putting the sphagnum in the endometrium and pulling it through the vagina is really the microbiome that contaminates the sample. Whether then the bench, the knife, the way that the sample is being cut to allow you to get a swab for microbiome analysis, you're just picking up the microbiome that happens to be lying in the laboratory bench, and then trying to find statistical ways to remove all of this contamination. And in those bars, the light green that you can see throughout is just areas that were contaminants. And if you haven't applied all the stringent protocols, you would have thought that this is the microbiome of the endometrium. But in essence, we found that we recovered bugs from 75% of our cases in the upper genital tract. There appears to be discontinuation at the fallopian tube and ovary level in 12% of the cases. And in also 12.5% of the cases, there was no association between the upper and the lower genital tract. We then took a group that had cervical cancer and a group that didn't have cervical cancer. And we found higher microbial diversity and lactobacillus depletion. And you can see on the left the bugs that are associated with endometrial cancer and the bugs associated with benign endometrium, again suggesting that there may be a role, but of course nobody knows if it's the chicken or the egg. And this is, I think, where it comes a very interesting topic that you can go on forever and do association studies and say, oh, you know, if you have cancer, you have this. But is it actually the cancer that causes these anaerobes to grow due to the tumor necrosis in the cells, or is it just the microbiome that eventually created an environment that promoted this cancer to develop in the first place? And this is where it becomes more complicated, more challenging, and of course, more interesting. How do you go from association to causality? And this is very difficult. The first thing is that you need, I think, to go back to longitudinal data from biobanks and see what actually happened in pre-diagnostic samples, if this is available to you. See what the microbiome was before the disease that you're looking at has been discovered, and see how the microbiome associated with the outcome that eventually came out. So in the cervix, which is the area that we moved in all of these steps that I'm going to go through in the next few slides, we went and collaborated with Anna-Barbara Mosiski, that many of you may know from UCLA in San Francisco in the US, the past president of the American Society of Colposcopy and Cervical Pathology. And Barbara had one of the biggest biobanks funded by NIH on serial HPV samples in young cohorts of girls that were beginning to get infected by HPV. And we went in a very precious population of women who had CIN2 diagnosed, and we looked at about 80, 90 women that did not receive treatment with loop excision immediately. So we went into these women's samples, and Barbara has published back in the days one of the seminal papers on not treating CN2 lesions and the regression and progression rates in young women. So we took the samples from that really interesting cohort of 87 women of young age, as you can see, that had histological diagnosis of CN2. And we looked at the 4-monthly samples that they had with liquid-based cytology. And we've done microbiome analysis of the baseline sample and all the 4-monthly ones that followed. We described what really happened in the microbiome of those individuals. And ultimately, we correlated the baseline microbiome, i.e. at time 0, to the chance of the CN2 progressing or regressing. And you can see on this slide that the lactobacillus deplete microbiome at time 0 had an adjusted odds ratio of 3.56 into predicting non-regression. So if you had lactobacillus depletion at Genera level, you had this odds ratio as a chance of non-regression, as opposed to somebody who was lactobacillus dominant, which is the comparison group. And at 24 months, it was the same pattern, slightly lower adjusted odds ratio, but still significant. Again, the numbers as we went to 24 months of return samples were smaller. And of course, this was adjusted for age, ethnicity, contraception, douching, smoking, and HPV 16 and 18. And again, a different way of demonstrating how the presence of CST4, which was depletion of lactobacillus, was associated with a slower clearance of CIN2, as opposed to lactobacillus dominant species. Now, this was also very interesting, but I think it still does not prove causality. It takes us a step closer to understanding that actually it is not it's not the disease that leads to the microbiome being different, but it's actually the microbiome that led to the outcome of the disease. The second most important thing in understanding causality is to understand the impact of treatment. And in this case, we're very fortunate because we have a surgical treatment for those patients that is indicated, and it's ethically completely appropriate to perform. And this is the loop excision. So what does the loop excision do to this microbiome is also a very important step into understanding causality. There was this group in China that looked at 26 women, and they concluded there was a shift for decreased diversity after you treated a woman for her CIN3. We've done the same in our institution, and we had about 105, 110 women, if I remember well, where we looked at the microbiome before the lip excision and the microbiome after the lip excision. And at the same time, we looked at a number of pro-inflammatory cytokines. And we actually did not find what the study from China has found. We found that the microbiome, despite removal of the disease, remained pretty stable. So a significantly higher proportion than women who don't have disease with CIN have lactobacillus depletion. And despite the fact that you took out their CIN or even cleared their HPV infection, those individuals continued to have increased depletion of lactobacillus, despite being even HPV negative in the subgroup we looked at. And we also found that their cytokine levels, inflammatory cytokines, continued to be raised 6 months after treatment, despite the fact that the disease was now gone. Which goes to show that possibly women that have a vaginal microbiome like this may probably predispose them to the disease, but the fact that they clear the disease does not revert the microbiome back to normal. So these individuals probably constitutionally, for whatever reason, have this microbiome. And it may be the reason that these individuals remain at high risk of getting the infection again and rapidly reacquire HPV. That's why these people are possibly at a high risk of getting cervical cancer again in the future. The next step in understanding causality is to explore the mechanism. And that becomes even more complicated because you need cell models to be able to do that, to understand how the lactobacillus may actually work in this setting. So what we're currently doing with Professor Dorber and Konstantinos, one of my fellows, is to try to use immortalized human keratinous cells, HPV infected. It's one of the models, the NICs, that Professor Dorber has been using for several years and developing for several years as a very good model for HPV from the foreskin. And in that, we are trying to do a number of experiments to understand how applying lactobacillus conditioned medium may affect the copy cell number, the live cell imaging, the E6 and 7 gene expression. And the same thing, this is my other fellow Nita, is trying to do in the endometrium, but in this occasion, trying to use organoids, miniature and simplified versions of the endometrium and ovaries that show a realistic microanatomy. And we want to inoculate with microbial products in order to assess the effects on those pathways. This is one of her organoids. It has been a major challenge to try to build this because this is a very, very novel but promising area, because you essentially take cells from the same patient and you actually try to grow these cells in appropriate conditions and keep them alive. And it has a great potential in the future into being something where you can use, for example, a chemotherapy agent and see whether the tumor cells are particularly responsive to that. So it's something that is extremely interesting for the future. But I think we're still at the beginning, and assuming that this model continues to preserve the same mutations, the same genetic instability is something that is currently being explored when comparing tumor to the organoids. But I think as we're going to go more into the future, I think we will see more and more of this being very, very interesting. The fourth and final step, in my opinion, is to see how you can bring all of this information into clinical practice. And this is where metabonomic platforms come into the mix. Platforms that may be able to be used to describe in bedside tests how the microbiome may be reflected in a sample that has been taken, and give a snapshot of the microbiome at a given time without having to send the sample for next generation sequencing and trying to do bioinformatics on an individual patient. And I think that at the same time, these techniques, metabonomics, have a great potential in cervical disease, because they're essentially a reflection of the balance in the whole of the biological system. They don't just describe the genetics, the epigenetics, the protein transcriptomics, but they bring into the mix the metabolites, those interactions that happen, and give the ultimate end product of all of these interactions. And we try to test different techniques that have been pioneered at Imperial College in cervical disease, for example, the eye knife, where in the diathermy, the smoke that is being produced when cutting a cancer tissue can go into a bedtime machine, and this can give you feedback on whether you've managed or not to clear the disease, and whether the cells that you're cutting are free of tumor. We try to do it with a laser in people who have intraepithelial lesions, creating an instrument that can give you at a specific point a replacement of the punch biopsy to allow see and treat at a single unit. And of course, same techniques using in liquid-based cytology cell pellets to see if they could even replace molecular markers with some funding that we have from Cancer Research UK. And I think these techniques will come and be very interesting as well into bringing the microbiome into the practice by being able to tell you what is likely to be the community state type in that individual as a microbiological marker without all this extensive bioinformatic analysis that is required. And I think as soon as we have all of these things clarified, which you can see is a complex process to go through, from just saying, oh, the microbiome causes it all, it's only then that after we understand all of this, we can think about diagnostic, prognostic markers, we can think about cause-directed treatments with probiotics, prebiotics, and even transplants of microbiome, as we're going to discuss in a minute. I think in the future, if we do understand these community state types, this microbiome that is associated with disease, we will be able to correlate this to the outcome and find that in pre-diagnostic samples, this makes a woman more high risk of not regressing, makes this woman more high risk of progressing, and be a useful tool in individualizing who we are going to treat or not. And I'm going to say a couple of words about probiotics before I finish, because I think it's a major area that has exploded when all of this microbiome discussion came into the mix. In many countries, you could just go to the pharmacy and see all of these preparations claiming to improve your gut, improve your health, your immunity, clearing your HPV virus, clearing your thrush and fungi. And without want to dismiss any of this, because it may be actually the case that they do work, I think the explosion of all of these preparations was probably based on the fact that these were not really regulated. And anybody could bring those to the market with very little evidence that they actually work, and even more little evidence that they do not cause any harm. And I think this is what the FDA and other organizations have recognized. And my understanding is that from 2021, these are going to be managed as drugs. So you have to go through drug trials to be able to prove a benefit of something before you're able to claim that it works as a drug to clear your HPV virus. And there will be more regulation, which will obviously also push the pharma companies to do the trials that we really, really need, because some of these things may actually do work. We just don't have trials that prove this yet. And of course, be very cautious that we don't have adverse effects as we do with all the other drug trials. In the area of HPV, there have been a number of small studies trying to use things like oral preparations that are, for example, in yogurt, and assume that by changing the gut microbiome, you change the rectal microbiome, And by changing the rectal microbiome, you also alter the vaginal microbiome. And although this is plausible in theory, I think we have no clear evidence that any of these yogurty type of preparations that are swollen and go into the gut have a direct access to the vaginal microbiome. And even the impact of the gut microbiome to the vaginal microbiome it remains relatively unclear. And this is another very interesting, coming to the end of my talk, concept from a group in Israel. And we all know that fecal transplants are practiced, and they have been introduced in clinical practice for people that have difficult to treat Clostridium difficile. And similarly with the vagina, there was a group with a small brief communication in Nature Medicine, where they found 3 donors of good microbiome and women with intractable BV that they have not been able to treat. And they've done what we call vaginal transplants to revert those individuals' microflora in the vagina to a healthy one. And although this was just a very, very small series, it gives you an understanding of how big this field is. But the steps have to be careful, because there is a danger into jumping into conclusions that and use preparations that may cause more harm than good. And I think pushing the pharmaceutical companies to run big studies, appropriately designed, randomized, controlling for quality, peer review, will help us to bring all of this medication in the future into clinical practice and see how we can find a preparation that will revert an abnormal microbiome that may make this woman more susceptible to HIV, to recurrent fungi, to preterm labor, to HPV, make this back into a Lactobacillus crispatus dominant microbiome, and also show that this is sustainable after we discontinue the treatment, because we really don't understand what creates the microbiome in the vagina in the first place. And I think that there's a lot to be done, but certainly it's an extremely, I think, interesting area. And I want to finish by making us all think that if you look at the whole of the history of planet Earth, and the little tiny fraction that the Homo sapiens and the humans have been on Earth, the time that the bacteria existed on this planet, it is very, very important to see how this evolutionary grew with different animals, species, and the humans when they appeared on Earth, and how there is no doubt that this play an important role into what happens in the human body. And I think we have, because of lack of ability, ignored for several years how important this is. I remember when I was a very junior academic, I heard a talk by one of our senior academics at Imperial when he was about to start all this project around the microbiome and the human body. And he was talking exactly about the example of 2 individuals that used paracetamol for their headache. And one of them, the headache goes away, and the other one, it does nothing. It's like eating a chewing gum. And the reason was because of the bacteria in the gut that were able to metabolize the paracetamol into the active form that helps the headache to go away. So what really controlled the function of one of the commonest and commonly used medication that we use was actually the bacteria, that may in turn be affected by genetics or just purely your diet. And I think it's very, very important to continue work in this field. So I'm going to leave you with Louis Pasteur and his words several years ago, and wise words that at the end of the day, it is really the bacteria that have the last word. Thank you very much.
Thank you so much, Dr. Maria. It was very interesting and informative lecture, and it has opened up quite a lot of doors with respect to what could be the possible cause, one of them being vaginal microbiome. And as a tumor microenvironment comes into play in all our carcinogenesis. So I would like to ask one thing, and that is, does it differ from, as you have shown, Asians, Hispanics, everyone has had different thing, different vaginal microbiome, little bit. Is it that is making us prone for various cancers and the differences in different regions? So you get cancer, cervical, like for us, we have cervical cancer, very common. Could that be a reason? And how do we go about collecting these samples, if at all we do a study?
Well, I think it would be a step too far to say that the microbiome difference in the CST4 by, I don't know, 10% or 20% is the reason that some countries like yours have a high rates of cervical cancer. I think the obvious answer to the high rates is the absence of an organized screening and vaccination program rather than the microbiome. But I think it is interesting to see and understand in the future what really makes some individuals, as with COVID, more susceptible to something like HPV. I think it's a fascinating area to understand. It's similar with HIV. You know, there are people who have sexual relationship with HIV positive people and they never get the virus. There are people who are couples and sleep in the same bed with somebody who's COVID positive and pre-vaccine, and they still don't get COVID. It is, I think, quite phenomenal to try to understand viruses and what makes people respond so differently to them. And I think there's genetic reasons. I think we just done one of the biggest genome-wide association studies in cervical disease that looks at some factors and some SNPs and polymorphisms that are more present in women for a cancer that is not considered to be hereditary, but it shows some hereditary elements in the way their immune system works and is able to respond to the HPV infection. And I think in the years to come, we will continue to understand more and more. And within those factors, I think microbiome is one of them, but actually, I don't think it's the only factor. I think it's such a complex system where you have the host and the virus and the bacteria, and different elements of this should be understood in order to understand what makes some individuals so susceptible. In terms of sampling collection, I think we can continue to collect samples, but there are already major biobanks of HPV samples collected, and sometimes it's probably easier to go to those and try to retrieve those samples and answer specific research questions, I think.
I thank you so much, Dr. Maria, again.
Thank you
for your excellent talk. There are no more questions, I think, from any audience that I could see.
Would they be in the chat box? Yeah.
Yes.
Well, thank you very much for the invitation, and hopefully next time we can also be there in person when COVID goes.
Yes. Very much looking forward to it. Thank you so much, Dr. Maria. Now, may I request Dr. Rekha to give the vote of thanks?
Yes, ma'am. Good afternoon, everyone. I think it was a very interesting session, and I take this opportunity to thank extensively all our eminent and distinguished speakers who joined us today. To begin with Dr. Priya, thank you, ma'am, for a clear description of the immune responses in HPV infection, and you showed us how different it is from the immunity developed from the vaccine, how the there is no, you explained very well as to how there is no inflammation when there is infection, but when there is a vaccination, the immune system takes over, and that is how it prevents in development of the cancer. And thank you also for so much for sharing all your extensive research work and explaining so clearly all the mechanisms, and also for giving us future research ideas that will be very useful for the juniors. And Dr. Smitha from our hospital, thank you, ma'am, for explaining all the various diagnostic tests and their pitfalls and advantages and what's new in the HPV diagnosis. Thank you so much for explaining everything. Dr. Jayashree, ma'am, and thank you so much for the extensive presentation on the role of estrogen in cervical cancer. We are always thinking of estrogen in endometrium, and you explained so very well about estrogen and possible role of progesterone receptor expression and future research possibilities in this area. And Dr. Ravi Diwakar, sir, thank you so much for explaining about or enlightening us about the immune therapy and the future of immune checkpoint inhibitors for recurrent cervical cancer, and detailing all the recent trials in that field. And most important of all, Dr. Maria, thank you so much for joining in from UK, and for an extensive elaboration on the microbiomes of the vagina and their interactions with the HPV and how it can lead to cancer. And it's also opening more doors to probably study how these microbiomes are causing cancer in, as ma'am said, in different regions of the world. Probably that will be an interesting area of study. And most importantly, I would like to congratulate Dr. Annapurna for bringing up the CME and inviting all the distinguished guests to enlighten all of us about HPV immunology, which is a very rarely discussed topic, I can say, in most of the conferences and CMEs. Thank you, ma'am, for giving this opportunity to speak, and thank everyone.
So to those attendees, I would like to make an announcement that CME certificates will be shared with the ones who have registered with their email IDs in the next 10 days. That this brings us to the end of the session. Thank you all for attending and giving a patient hearing to us. We from Shankara Cancer Hospital are indebted to all our speakers who have shared their vast knowledge with us and made us more knowledge rich. Thank you so much once again.
Ma'am, shall we end the meeting?
Please, we can end the meeting. Thank you all.
Thank you, ma'am. See you.
Thank you.